Hi everyone, and it's great to see you and to join us here. This is the first session on coral reef, and so I have just a few words first to thank people who worked on reviewing on all the papers. So this session on coral reef will be with Nicole, Alexandra, Cloé, Luna. We'll start with Nicole and Alexandra before having a break. We will go 2 times in the Philippines, one in Cayman Islands and one in Colombia. Nice. I will thank all the reviewers of the papers. Generally speaking, it's 22 reviewers and jury members who spent more than 100 hours to, at least we paid them 100 hours. I don't know how much they took to review all the papers. To realize it's important in terms of work for us. So I'd like to thank Dominique Adriens. So those are the reviewers of this session. Dominique Adriens, professor at Ghent University. Aldine Abiel, research engineers at IRCAN Université Côte d'Azur, also part of the jury today here. Hello. Steve Como, researcher at Sorbonne Université Love. Florence Lacrosse, project manager at Coral Guardian. And here behind me, hello, Florence. And Aziz Moula, postdoc at Ecosys Université Côte d'Azur, also here. They cannot see what's you type on your screen. And Brian von Herzen, executive director of the Climate Foundation. So, for the jury member, it's going to be Aldine, Florence, Aziz, and me. I'm mostly the chair. Okay. Remember, it's 15 minutes for the presentations. After that, 20-minute discussion. We can start with the jury, and then I'm sure you will have questions. Of course, good luck with the heat. For those of us who are not following us online, it's a heat wave also here on the Côte d'Azur, and the air conditioning is quite limited. Are you ready? Nicole, c'est parti. Right.
To switch the slides or
Yeah, don't worry. Right. Let's take a few seconds to show you that. We just do that. So, as usual, you have that, the exact same that you had for 2 years. And so you should be able, yes, to change that. No spoiler. As usual, if you want to friend, a family to see you, it's best to stand in the frame of the video. It's more here. We can then adjust a little bit if you want.
Okay.
All good?
Yeah.
Do you have any questions before we start? Not for you? All good? The 15 minutes are fine. I need of a timer.
We'll see.
Allez. Let's go.
Okay. So hello everyone, my name is Nicole, and I was working with Blue Alliance and with the University of Constance for the last 6 months, and today I will be defending my thesis titled Daily Temperature Variability Promotes Resilience to Acute Heat Stress. So in order to introduce this topic, I want to take the time to know that ocean warming has become one of the main concerns for the reefs in the future, and their decline will depend on the capacity of these reef organisms to adapt to these conditions. Now, on this, you can discuss that corals are keystone species in these ecosystems, and they play an essential balancing role. However, they're also highly sensitive to ocean warming due to their symbiotic relationship with zooxanthellae. And unfortunately, we have been seeing more frequent heating events. In the last bleaching event, over 80% of the reefs bleached, and we only expect the situation to get worse. This being said, it is crucial to identify naturally thermotolerant corals, because on one side, it's going to help us characterize environmental parameters that could be related to resilience and therefore incorporated into conservation efforts, but also find locations that could serve as potential thermal refuge. Now, also important, we want to distinguish between coral bleaching and thermal threshold. So on one side, bleaching is the process where corals expel their zooxanthellae, and therefore that causes their tissues to appear white. Therefore, this is a stress response, and yes, it can be caused by temperature, but it can also be caused by pollution and sunlight. On the other hand, a thermal threshold is the temperature above which rate starts to decrease, This is therefore related to metabolic rates such as photosynthesis. Now having these two definitions, we can jump into the key concepts. So the first one is thermal tolerance, which is the ability of an organism to withstand variations in temperature. We also have coral bleaching, which is the ability to withstand the bleaching. And then finally, we have resilience, which is the ability to recover from a disturbance. With these concepts, then I want to introduce a little bit about the area of study. This was the Verde Island Passage. It's located between the Luzon and Mindoro Island in the Philippines. It is a vital corridor within the Coral Triangle, and they have described over 300 species of corals, as well as the 60% of the known shore fish. Not only is it special, but it was also postulated as a potential thermal refugia 3 years ago, because using a high-resolution ocean simulation, they predicted that this place had high daily temperature variability, probably caused by tidally generated internal waves. This, and last year, Blue Alliance was doing a monitoring during the bleaching event, and we observed a low percentage. So taking these two together, we came up with the research question, which was, how does coral thermal tolerance and resilience vary between reefs that are exposed and protected from the Verde Island Passage? And to break this question, we did 3 different objectives. The first one was to characterize the thermal parameters. The second one was to determine the thermal tolerance to acute heat stress of Posilopora. And then the third objective was to identify thermal history parameters that could explain the variability in that tolerance. And how did we do this? So the methodology is split into 4. We have choosing the area of study, characterizing the thermal history parameters, doing the coral sample collection, and then taking those corals to the lab, which was the coral bleaching automated stress system. So for the area of study, we have Puerto Galera, which is in Mindoro, so south of the Verde Island Passage. And it was just great because we have 2 protected and 2 exposed stations very close to each other, and that made it quite easier to collect in both. And this study took place between March and May of 2025. For the temperature collection, we placed HOBO sensors in each station. They have a resolution of 0.01 degrees Celsius, and they registered temperature with an interval of 2 minutes. These were placed between 9.5 and 10.5 meters. And then we calculated the thermal history parameters with an accumulation window of 4 weeks. This means that we look at 4 weeks of history of the temperature. And for the purpose of this presentation, we keep heating as positive values and cooling as negative values. Now, for the thermal history parameters, we divided them into 2 categories. The first one is the cumulative thermal stress, and this is a deviation of the MMM, which is the maximum monthly mean. In this area in the world is 29 degrees. Using this, we calculated the degree heating weeks, which is widely used in coral research. And we also use a degree cooling week. So it's flipped. We look at temperatures below that MMM. Now, on the other hand, we also looked at daily temperature variability. And we took the mean daily temperature range of the last 4 weeks from that date. So for the coral sample collection, it was very interesting and challenging. We used Posilopora because it was the most abundant genera within the stations. And we had 10 tagged colonies per site that we would look to afterwards. And they were found at depths between 8.5 and 13 meters. And every date that we were collecting, we would go to 1 exposed and 1 protected site. We did this for 5 dates, 3 in April and 2 in May. And we did it with scuba between 7 a.m. and 11 a.m. Then when we had the corals, we would have to run all the way to the lab, which was a coral bleaching stress automated stress system. This is a rapid diagnostic tool to identify thermotolerant corals. And we use acute heat stress because it's short and strong heat. So it lasts 24 hours, and it consists of 4 independent tanks. We have a control tank, which has a constant temperature of 28 degrees. And then for the other 3 tanks, we have a heating period of 7 hours, and then they also stay at 28. That means that we have a heat stress period followed by a recovery period, and the tanks go at different temperatures. So this tank went to 32 degrees, this one went to 36, and this one went to 40. And we also turn on the lights and turn them off in order to follow that normal diurnal cycle. This being said, what we also measure is the dark acclimated photosynthetic efficiency, which is a proxy of bleaching. We do this at 7 hours, 18 hours, and 24 hours. Therefore, the first two measurements are related to the tolerance, but the last measurement, since it's after the recovery, it is giving us an indication of resilience. Then for every time point, we determined the thermal threshold, which is the ED50. This is the temperature at which that photosynthetic capacity is reduced to 50% of the original value. Therefore, a higher ED50 means higher thermal tolerance. Now let's look at the results in the discussion. So the first thing is the in situ temperature. So we have here an exposed and protected site, and this dashed line is showing us the temperature mean of the entire period. And we can see that they're very similar. However, what does change is that the daily temperature range in the exposed sites was higher. And not because the maximum was different, but because the minimum was different. Now, we can also observe that these temperature drops were more intense during new moon phases, and that is what is telling us that this is likely caused by tidally generated gravity waves. Then with the temperature, we looked into the temperature parameters, right? So every point that we look at here is referring to 4 weeks of history of that temperature. And between exposed and protected sites, we saw that there's a similar cumulative thermal stress. So for degree heating weeks, which in this graph is in the dotted line, it remains 0 for the entire period because we never had temperatures above the 30 degrees. So you can see it there. However, the degree cooling weeks actually decreased during the study period. Again, they decreased because we look at the absolute value. So they went from -10 to 2, and yeah, it's flipped. And even though we saw similar cumulative thermal stress, it wasn't the same story for the daily temperature variability. We observed that this daily temperature variability was higher in the exposed sites, which are in green, compared to the protected all over the study period. And then knowing these temperature parameters and knowing the thermal tolerance of the corals in the time of collection, what we tried to do was relate the thermal tolerance with those degree cooling weeks. And we performed a correlation between these two variables. So here we have degree cooling weeks. On this side, we have the thermal tolerance, which is the ED50. And the first thing is that at the first two time points, so 7 hours and 18 hours, we observed a strong positive correlation where we have an increased thermal tolerance when the cumulative thermal stress is decreasing. Now, what this suggests is that degree cooling weeks is inducing an acclimatory response. And it's aligned with previous studies that also saw that degree cooling weeks can change this thermal tolerance. However, there's this last article from Roth in 2012 where they compared heating and cooling, and they found that heating is ultimately more deleterious, and that's probably because the temperature optimum of corals is near to the MMM. Now, even though we saw this in the first two time points, this is the time point that was fascinating to observe because the pattern completely changed. So at 24 hours, we observed that the exposed and protected stopped following the same trend. So the protected sites, so the ones in orange, have the same trend compared to 7 and 18, but the exposed sites remained constant. The ED50 doesn't drop, even though we have a high cumulative thermal stress. And this raised a very big question, and then we tried to incorporate a new parameter in order to explain this difference. And we did this with a mixed linear regression with an interaction using degree cooling weeks and daily temperature range as explanatory variables for the ED50 at 24 hours. The results from this regression were an adjusted square of 45.5% and an accuracy of 90%. And in this graph, again, we have the thermal tolerance and the degree cooling weeks, but the different colors are different values of daily temperature range. So the dark blue is a higher daily temperature range compared to the lower one. And we observed that if we have a higher daily temperature range, then we always have a higher thermal tolerance. Now, what is really interesting to look into is that this difference is higher when there is a high cumulative thermal stress. And that is why this difference is higher on the left side of our graph compared to the right side of the graph. All in all, this what tells us is that it's not the magnitude of that daily temperature variability that is explaining the thermal tolerance, but rather how that interacts with the cumulative thermal stress. And at the end, what we can wrap with the model is that the effective degree cooling weeks towards thermal tolerance at 24 hours is explained by that daily temperature variability. And this suggests that daily temperature variability may mitigate the effect of cumulative thermal stress on thermal tolerance at 24 hours. And that's why we suggest that it promotes resilience, because this time point was after the recovery of a heating stress. And this observation was aligned with previous studies that found that daily temperature variability reduces the risk of coral bleaching. Now, the mechanisms that can lead to this are different. We could have adaptation, acclimatization, or environmental stress memory. But from what we did, we can't really conclude into what mechanism is doing here. And as much as these results are very interesting and very fun to look into, it is important to consider that whether these results translate into a higher thermal tolerance or resilience during a heating event in these exposed sites is a question that remains open. Now, even though we looked at temperature, we know that non-temperature parameters are also important. And those that could be related with the internal waves are, on one side, the availability of nutrients and plankton, because a heat stress causes a higher energy demand, and therefore having a reserve could promote resilience. But on the other side, these places also have an increased water flow, which actually can remove those harmful pollutants and therefore decrease the oxidative stress. And all in all, further research is needed in terms of water quality, and that this process is probably species specific. So we need to do it a little bit more wider. Now, looking into the conservation potential of all of this, even though these corals could be thermotolerant, they're still vulnerable to other kind of stressors, such as overfishing and pollution. And therefore, it would be both relevant and beneficial for daily temperature range to be incorporated into marine spatial planning, taking into consideration that internal gravity waves might imply a trade-off for the corals. So they might be more thermotolerant, but maybe they grow slower. And finally, also important is that the relationship between thermal tolerance to acute heat stress and bleaching resistance is still an association that needs to be further explored. So in order to conclude, we go back to our research question, which was, how does coral thermal tolerance and resilience vary between reefs exposed to and protected from the Verde Island Passage? We achieved our 3 objectives. The first one, we observed a similar cumulative thermal stress, but a different daily temperature range in the exposed sites, where we observed a higher daily temperature range in the exposed sites. Thank you. And then the second objective was the thermal tolerance. We observed similar thermal tolerance in both the 7 hours and 18 hours, but we observed a difference in resilience, where the exposed corals were more resilient compared to the protected ones. Our third objective was to integrate these parameters, and we observed that by taking both cumulative thermal stress and daily temperature variability, we could increase the accuracy of evaluating coral resilience. And to answer the question, under cumulative cooling stress, Posilopora from exposed reefs to the Verde Island Passage have a higher resilience to acute heat stress compared to those that were protected. And to finish, this study wants to highlight the importance of incorporating additional thermal history and environmental parameters beyond cumulative thermal stress. Thank you very much.
Okay, congratulations also for timing. Who would like to start? Adim, Aziz? Aziz? Okay.
Okay, cool presentation. Very good. Much better to watch than to read. I'd say.
Glad to hear that.
My first question is, you kind of answered it in slides 11, but I kind of want a bit more detail. Why Posilopora? I don't like the idea of it's just abundant. Like, I want to know why this particular genus. Why not the other 270 species you have there?
Okay, so what we observed is that, like one of the important things is that since it's the more abundant, it's probably giving us a little bit more of information of what is happening in these reefs. And also what results quite interesting is that this genera is mainly photosynthetic. It's not that much heterotrophic, and therefore switching into an heterotrophy isn't that easy for them. And then that daily temperature variability could actually be related to the coral bleaching and to the thermal threshold. So that is why it was also relevant to choose that species.
Okay.
Well, the genera.
And it is it's the most abundant in the
Yes.
area. By what coverage are we talking about?
I can't tell you exactly, but from observation,
Yeah.
I think it's probably at least 30 to 40% of the corals that we find.
How many species of Basilophora are there in this complex?
So it's actually quite challenging to distinguish between species of Basilophora. Again, from observation, we saw Damicornis, we saw Verrucosa, and we also saw Meandrina. And it was quite challenging to know if it was or not Verrucosa or Damicornis. We could think that it might be Verrucosa, but we would need to look into the genotype and more information in order to confirm it. But it's probably those three.
And my final question on Basilophora is there any difference in the thermotolerance between these species?
From the research and the literature review, I observed that a lot of people used Basilophora damicornis, and the results that are reported are with damicornis, and I didn't find any comparison between Verrucosa and damicornis. However, since Verrucosa is a little bit more thicker, maybe they could tolerate more, but it's still a very open question, and I couldn't really answer what would be the differences between the species.
Okay. So I can't remember which slide it was where you were defining thermal tolerance variation in temperature. There was a slide where you were defining this.
In the introduction?
Maybe not in control of it. Already, the Arabian Gulf has many corals which survive 32, 33, 34 degrees, and you tested 40 degrees. Is this realistic, or is it just an extreme you wanted to test? What was the reasoning behind that?
Okay, I'm going to use one of the annexes to explain your question. Can you answer? See, it takes a little bit of time to.
It is going.
Yeah.
Yeah, it's good.
Just give it some patience.
It's only the screen width, which is.
So basically, when you do a sea bass, you want to have very low temperatures and very high temperatures because we use an exploratory curve. And that's the image that I'm going to try to find here, but here you go. Sorry. Here, this one. So when you do the coral bleaching automated stress system, you want to have temperatures for this entire curve, right? Because you want to find where that photosynthetic efficiency is at its maximum, but also at its lowest. Because when we have this logistic regression, we find, so if this was the 100%, we look at the 50%, we go here, and then we find the temperature. So if you don't have that full curve, then you can't really have a higher confidence towards it. You don't know if the curve was actually dropping or not.
Okay.
So yeah, that's why we had to choose those extreme temperatures.
And so this region, the protected exposed sites, one can have thermotolerant corals and one maybe not. Are there any other areas in the Philippines or in the world or in the region that have been identified as thermotolerant corals, apart from in a lab?
Yes. So this is a very new topic, and there's not a lot of research in it. There's only one paper published by Dr. Wall in 2015 about internal gravity waves and bleaching resilience. That was in the Cayman Islands. However, the research from Bachman and collaborators in 2022 used this ocean simulation. So it's not a pinpoint in one side, but rather with satellite data and a little bit of theory of how when you overlap internal gravity waves, but also the mixed depth layer being shallow, then you can have that daily temperature variability. And they use this figure to explain it. That's why I put it, because I think it's really good. So in scenario A, you have these waves, but there is no variation in temperature because the mixed depth layer is too deep compared to where those internal gravity waves are happening. But in scenario B, if that mixed depth layer was actually shallower, then we also have that daily temperature variability. So there's not a lot of stuff pinpointed, but they found different places around the world, mostly in Galapagos and the Coral Triangle.
Okay. So the topography is as important. Okay. This is. Can we ask questions on the thesis?
Yes.
Yeah. Okay. In your thesis, I forgot what you were talking Sorry. Let's go for this one first. You mentioned the extent, maybe you don't remember, but I'll read it to you. The extent reefs decline will largely depend on the ability of reef organisms to survive under warmer conditions. Like I said, they already survive under warmer conditions in the Arabian Gulf. So what else is important for them to survive in the future?
Well, as I mentioned previously as well, there are other kind of stressors that are being increased, right? So we have pollution, we have ocean acidification as well, we have overfishing. So we need to remove those stressors in order to also promote these thermal tolerant regions, right? These potential thermal refugia. Because if we don't control those stressors, then it's not really gonna work. And I think further research needs to be done into seeing if these corals are indeed or not more vulnerable to ocean acidification, which is definitely also going to increase, but also pollution.
Yeah. One thing you didn't mention in your thesis or talk was reproduction.
Sorry?
Reproduction
Reproduction.
as well. Would you not say reproduction is more important than just survival, the measure?
Can I have access to my slides, please?
Yes.
Sorry. So there is a relevant study that I very briefly mentioned, and it's this one. So Putnam and Gates, what they did is that they preconditioned the adult, and then they looked at that effect on the offspring. And they saw that there is probably a transgenerational acclimatization activity going on, and that when the parent is being exposed to heat, that acclimatization is actually transferred to the offspring. So for sure, there's more research that needs to be done, but this might even have a good effect in reproduction as well.
Because even if the corals didn't show any signs of bleaching, doesn't mean they haven't been impacted as well. So do you have any data on this, previous bleaching events in the area where reproduction has been affected, or you didn't?
So
because you were there during reproductive season.
Well, one of the things here specifically for this area is that we are still not sure when the corals are spawning and how they're reproducing. To be completely honest, the coverage is just so beautiful and it's full of stuff that I didn't even find recruits. So I couldn't tell anything about how this might impact the reproduction there in that site in specific.
After is it?
Yeah.
It's fine.
Yeah.
I did.
Okay, so thank you so much
Thank
for
you.
this really complete and nice presentation. So my first question is, do you know if coral diversity can increase the dermatology in the reef?
If coral diversity can, as the ecosystem in
Yes.
full? Well, I'm not sure if the ecosystem fully can increase it. Now, we know that some genera are probably more thermal tolerant to others, and also some genera have a higher bleaching resistance. So probably the way you shift that diversity, if you have more genera of the ones that are resistant, well, the whole ecosystem will therefore be resilient. But what the effect of one persistent coral might be being right next to a susceptible coral, I didn't find any information related to that.
Okay, and then do you think that the resilience, so the resistance, is coming from the host or the sunlight?
I will use one of the annexes as well to explain this. So one of the things that I had to mention very quickly were the possible mechanisms here. And there are three. So we have adaptation, which is determined by the genotype. We also have acclimatization, which is basically how plastic that genotype is, right? And then the last one is environmental stress memory, which is basically you put a priming stress, let it recover, and then you have a final one. And actually looking into this needs to be done with the coral host, with the zooxanthellae, and the microbiota. And if it's adaptation, then we're going to have some results for conservation efforts. If it's acclimatization, then we have very complete different ones. For a paper of environmental stress memory, what they did do is they kind of simulated what would happen in those three. So what would happen in the coral host? This priming exposure will probably trigger epigenetic responses. Then on the other hand, for the zooxanthellae, then the triggering exposure might diversify the zooxanthellae into more tolerant ones. And then for the microbiome, same. We're going to have probably a little bit more thermotolerant microbiota. So it's probably the combination of these three and the interaction.
Okay, so then do you think now with all your study that thermotolerance is not only coming from, so the thermal resistance is not only coming from the acclimatation of the coral itself to the heat, but also combining with all these aspects.
Yes.
How do you envisage
well,
the next study? So how will you envisage the next study?
So yeah, exactly. With what I did, I lack the genotypes and the phenotypes, and assuming that funding is not a problem and the time is not a problem, well, one, I would incorporate all of these parameters that we could consider: water quality, plankton, nutrient supply, and I would look at not just the photosynthetic efficiency, but also at the zooxanthelle density, at the catalase activity, at the glutamine synthase activity, before and after heat stress, and looking at the protein signatures before and after the heat stress, because if we know what genes are being expressed before the heat and after the heat, then maybe we can start pinpointing what mechanism it might be.
Okay, thank you. Maybe the last one. How can you explain, your finding, you mentioned that under cumulative cooling stress, so Bacillopora from exposed reef have higher resilience to acute thermal stress compared to those found in protected reef. So can you explain this? How they can, at physiological level, how cellular level, how do you think they manage?
Wait, can you repeat your question? Sorry, I
So,
didn't really understand.
at one point in
I think it's
one
the
of your main
conclusion.
yeah. How can you explain that under cumulative cooling stress, Bacillopora from exposed reef have higher resilience to acute thermal stress compared to those found in protected reef?
So it's okay. So it's not really
The first one.
that the Bacillopora in that cumulative cooling stress are more thermotolerant. It's that this cooling period is a stress. Like if it's extremely cold, it's also stressful for the corals, right? And that it's the daily temperature range that makes the difference on their response to that. So it's not only the coral changing the thermal tolerance within the degree cooling weeks, but it's the daily temperature variability making the difference on how it shifts within that stress.
So do you think that any kind of stress can induce such resistance?
Well, we looked into degree cooling weeks, which, I mean, everyone has always been using degree heating weeks, right? And that is the measure that everyone uses. But when you really think about it, corals have a temperature optimum. That means that if you shift them towards heat, they're going to drop their performance. But also, if you shift them towards cold, they should be going down, right? So what we saw is that when we take them into cooler moments, they have that lower thermal tolerance. But if that translates into them being resilient to other kind of stressors, such as degree heating weeks, or maybe even pollution or overfishing, well, that was still an open question that we would need to look into.
Okay, thank you.
Thank you, Aldi.
All right. So let's go to you, Florence.
Yeah, thanks for your presentation. My first question is related to the methods, the sea bass methods in particular. I'm just wondering how you determined the limit between the tolerance and the resilience threshold.
Okay, so I will again use one of the annexes in order to explain this. I'm sorry, thank you very much. So there's a little bit of a story here that we weren't we don't have the time to explain it that much, but the original sea bass goes only 18 hours. And that means that it only had 2 time points. So there is some data previous to this, and we were seeing that the thermal tolerance at 7 hours and 18 hours was similar, but one was dropping a little bit more. And then that's why we incorporated this 24-hour measure, because we wanted to leave a little bit of a longer recovery period to see if it was changing or not. And to our surprise, the protected sites had dropped thermal tolerance in the first 4 days. And this was fascinating. And that's when we realized that we needed a little bit of a longer recovery period. And that's why we refer to this one as resilience.
Okay, I see. And is that based at all? I mean, there you talk about seeing in practice what's happening. Are any of the values based originally on the literature, I guess? This original sea bass method, do you know who that was developed by?
Yeah, so the sea bass method was developed by my supervisor, by Dr. Christian Bolstra, and some people in the USA as well. And that's how we chose the values. So before doing this, we did a little pilot of finding those temperatures for the full curve. And it was a bit, yeah, it was like, are you sure that this is going to work at 40 degrees? And he was like, yeah, well, if you say these corals are actually thermal tolerant, it should. And when we did that, we found the full curve. So it's kind of a little bit of a trial and error according to the site. I know in Egypt, in the Red Sea, they use it a little bit cooler compared to what we did here.
Yeah, okay, great. Great to hear. Regarding the non-thermal variables that you said also influence the stress, such as the gravity waves, were they all, you mentioned various studies that considered these variables. Were they all considered independently, or were they sometimes considered collectively, maybe with temperature as well?
Okay, so from what I have read and looked into the literature review, there's not many that do that interaction. And the only paper that looked into specifically internal gravity waves affecting bleaching, in their case, not thermal tolerance, was the one from Wall, and they only measured temperature. So even their conclusion at the end was also like, well, this is really interesting, but we also need to start looking at these parameters full. And I think it is a very valid and open question. And that's why at the end, we just highlight, like, yes, we need more thermal history parameters, but also environmental ones, because it's definitely going to play a role there.
Yeah, do you have an idea of which parameters you prioritize to look at collectively?
I think I would probably look at pollution indicators and water quality. I think that is the one that I would prioritize, and probably also turbidity, because since we have this tides changing, probably the turbidity could be changing as well.
Do you have an idea of what, like, do you know why you'd put pollution and turbidity? I mean, turbidity you just said, but pollution, why you'd put that one first as well?
So the thing is that corals are also bleaching, not with temperature, but also with different pollutants. So if we want to look at the effect of bleaching, well, we also need to incorporate those stressors. So that's why I would first include pollution, because it also causes bleaching. And then followed by that, I would look into turbidity, because it affects the sunlight, which is also stressful for the corals.
Okay, great. I have one kind of final question regarding the societal impacts of your findings. Do you have any recommendations based on what you found for the management of the Verde Islands passage?
Well, here, I think still this needs further research, but I think it's crucial to understand that thermal tolerance doesn't mean that the corals are resistant to everything. And specifically, Puerto Galera is a place with a lot of diving activity with tourists. There's over 70 dive sites here. So I think that if these corals actually grow slower and are not as big as other reefs, well, we need to start regulating it, because maybe when people hit them or kick them, well, that is not really good for them, because they will take a very long time to recover. So I think that is something that I would regulate, because overfishing has already been stopped in this area.
Okay, great. Thanks a lot, Nicole.
Thank you, Florence.
Thank you, Florence. So our jury members have been quite proactive in questions. So, but we have a question for a couple, time for a couple of questions on your sites. Oh, you have a question.
You mentioned at one point that because they have an increase in nutrients and all of that, that they might be more resilient, if I remember right.
Mm-hmm.
But if I remember right, corals are oligotrophic, right? So they prefer low nutrient environments. So don't you think they might also be outcompeted if nutrients increase by other fast-growing species, just as macroalgae?
It could happen, yes, and that's why more research is needed. However, there are some articles that started to look into the lipid reserve of corals before the heat stress, after the heat stress, and they compared the corals that were resilient and the ones that were susceptible to this bleaching, and they noticed that the lipid reserves were higher in the ones that were able to tolerate this heat stress. So I do think that this nutrient availability will probably help them instead of decreasing it. But of course, if you arrive to a point where it's just too much, well, they will outcompete, and then you're going to have a lot of algae in the ecosystem as well.
Okay. What about the discussion? Yes, Margot.
No, please go ahead.
It's just a quick question, and sorry if you said it and I missed it, but I think you said you put the temperature and you, I guess, also hard to see the coral between 9.5 and 10.5 meters.
Yes.
Why that depth? Because that's quite a small range. Right?
Yeah.
Is it
Just repeat the question.
Okay, so why did we put these loggers at that depth, right? So the thing is, between 0 and 5 meters, because of sun exposure, it's just too variable for us to see that depth. And also, probably because these internal gravity waves are coming from the deep, they're not probably achieving this depth. Still, it would be something that would be needed to look into. And going any deeper, it would have been also less safe for us as divers. The collections were very long. I think our longest dive was probably like 1.5 hours. So that is important. And also because most of the corals are there, we rapidly assessed the depths at which the corals were, and it went from around 4 meters all the way down to 25, 26. So that's why we kept it in the middle.
Thank you for questions, for your answers. You can have a look to this screen or the one before. So Fabian is saying hi to everyone. We've got Andrea saying hi. Hold on, there is a few. Hassan, Cassandra, and as this was before, during, and still during and after. Chris.
Thank you, Chris.
Very interesting. And oh, Jacob, nice picture, Jacob. Andrea.
Thank you, Hannah.
And Anna, but you're continuing online.
Thank you, Johan.
And Erika. So
Gracias,
congratulations.
Erika.
And so. And Chris is also saying thanks.
Thank you, Chris.
Okay, here we go. Ali, are you ready?
Yes, we're ready. Okay, let me get the timer though.
Yeah, I'm ready also with the keyboards. Oh.
Okay, I'm gonna sip the water first. Okay, ready? Good? Okay, hello everybody. Bonjour à tous. Okay, so today I'll be going over my master's thesis, which is called Stony Coral Tissue Loss Disease and Coral Community Change in San Andres, Colombia, Species Level Impact and Stakeholder Engagement. So, as we all are aware, coral reefs are incredibly important ecosystems. They're critical for biodiversity, coastal protection, tourism, and local livelihoods. 1 billion people benefit either directly or indirectly from this ecosystem services around the world. But unfortunately, like Nicole has just mentioned, they are decreasing at an alarming rate, and we're seeing this widespread. This is because the corals have a symbiotic relationship with an algae called zooxanthellae that lives inside of their tissue. The zooxanthellae gives them their nutrients, and when the coral is stressed, it expels the zooxanthellae and then it causes bleaching. So what can cause this stress? Like Nicole just mentioned, thermal stress can be caused, and this is we're seeing a lot because of climate change. Also, anthropogenic point sources of pollution and diseases, just to name a few. So if this circle represents how many coral reef-building corals there were in the 1980s in the Caribbean, this is how many we have left, 20%. If this circle represents how many of our tropical corals are protected worldwide, this is what is unprotected. So zooming in more on the Caribbean specifically, like I mentioned, coral disease is one of the reasons why corals are dying. The Caribbean is a hotspot for coral disease. One of these diseases is called Stony Coral Tissue Loss Disease, which is very long, so I'll be calling it SCLD for the next time. And this was first seen near Miami, Florida in 2014. So SCLD has spread across the Caribbean. It's now present in 28 different countries and territories. The exact pathogen that's causing this disease is not exactly known yet, but it's assumed that it is biological or that it's bacterial, sorry. It is very virulent, it spreads really widely and fast and causes widespread mortality. So zooming in further, in the Caribbean, looking at San Andres, is in Colombia, is home to the third largest barrier reef in the world and has 77% of Colombia's coral reefs. So with all this said, this brings us to our research question. How is skilldie affecting coral cover and species composition on San Andres? It is hypothesized that the disease prevalence will negatively affect total coral cover and will cause species shift in composition. But we're not just looking at ecological monitoring. I'm really wanting to look at social insights as well to extend the ecological change and also have opportunities for strengthening co-management and community engagement. And it's really the goal to provide actionable recommendations that support a more resilient and better protected reef system. So like I mentioned, we're looking at San Andres. San Andres is Colombia, but it's actually closer to Nicaragua here. And so we're looking at two sites mainly to look at the disease. Here we have Plaza on the northwest of the island and Nirvana on the southwest. And just to mention, this was done with the Perry Institute for Marine Science. Collaborators are seen here, and it's funded by Cordat. So San Andres is within a marine protected area. It's within the entire sea flower biosphere reserve. It's located here, in the southern section. It's the largest in the wider Caribbean region, 65,000 kilometers squared, and has a variety of zone types that you can see here. And I just want to note that Coralina is the regional autonomous government agency, which has authority of the environment. It's not the national government itself. So how are we tracking coral coverage? So PIMS uses photomosaics. And so here you can see a diver taking a photomosaic, and it's really following this plot here. So here's the layout. So in the plot, you have 4 different corners and markers placed, and then they're going north to south and then east to west to really make sure you have full comprehensive coverage of the entire area. Then this is where I come in. So the photomosaics are uploaded into a software called TagLab. This is kind of like the interface and what it looks like. And basically over the image of the photomosaic, I'm tracing the corals and assigning it a species. And then it's given the area and the perimeter, and then I can overlay that on the next month to then make a comparison between the two to track changes in sizes, regrowth, and mortality. And so you can see the months that were chosen to look at Nirvana and Plaza, and then all of this is exported as a CSV, so then we can do fun statistics. So for the social science aspect, I interviewed 32 different people from 6 different stakeholder groups. So we have the government, which is Coralina, as mentioned, NGOs, tourists, locals, divers, or dive operators, I should say, and fishers. And so there was the same questions for all of them, but also some of them changed depending on their expertise. So to jump into the results. So here we can see percent coral coverage. So Plaza is in the bluish color and Nirvana is in the orange. And so we can see that they both drop significantly. And a statistical test shows that they're actually not significantly different. Then looking at species level composition. So I'm just going to really pay attention to 2 species here for the sake of time. But we can see at the beginning of the analysis that Nirvana was really dominated by Orbicella annularis, where towards the end, this is really no longer the case. It is almost equaling the amount of Parietis asturitis, which stays relatively similar throughout the entire time. The same thing can be said at Plaza, but instead it's Orbicella fabulota, which was really the dominating species, and then again drops actually lower than Parietis asturitis, which really again stayed relatively similar throughout the year and a half. So then looking at colony fate by species, so it was found through statistical analysis that there was significant difference in the species that were looked at and the mortality. Here we can see the majority of them died. But post hoc Dunn's test showed that Favia fragum and Montestrea cavernosa lost 85% of their colonies, which was significantly more than Orbicella annularis, and Orbicella annularis was significantly more than the species listed there. This cannot be said the same for Plaza. Here, there was no significant difference between the species mortality, so widespread disease mortality throughout all species. So to discuss. So both sites show similar impact from sternocorial tissue loss disease, since we didn't see much of a significant difference between them. And this coral loss range is congruent with what other studies have seen. It's really highlighting a pattern of rapid, high magnitude coral loss associated with the disease. At Nirvana, we saw that there was mortality in the Fabianae genus, and this is where we saw Orbicella nularis and Fabulota, which is congruent with other studies. But we saw that F. fragum decreased a lot, significantly more than the others, and this is a little bit different than what other studies show. So maybe this signals potential local co-stressors. More studies need to be done to know for sure. But the collapse of the M. cavernosa is congruent with what other studies are showing. We're also seeing a big decline of these reef builders to a rise of weedy corals. And this is what we saw at the beginning with that 80% loss since 1980. And so, yeah, there's this big decline of the Orbicella species. So, and only this minor decline in the Parietes astratus is suggesting a potential relative resilience. And this is congruent, again, with what other species or other studies are showing, that stony coral tissue loss disease really restructures the reef towards these more weedy species, which Parietes astratus is one of. And so this could be reducing habitat quality, lower biodiversity, weaken long-term resilience, and have whole ecosystem level impacts, but obviously more studies have to be done in order to know this for certain. At Plaza, there was a broad-based coral mortality. There was no significant difference between the species. And so this might indicate a few things here, but I want to pay attention to the non-selective environmental stressor. Here is a map, again, of the island. And this part up here really shows you where the urban area of the island is, and this is where Plaza is. So on San Andres, there is no wastewater treatment facility. So they have untreated sewage outflow that's being pumped off of the west of the island, just offshore of the urban area, 300 meters or so offshore. And so that definitely puts Plaza in a place that is probably being affected by this plume. And it's been shown in studies that there's higher disease prevalence in sites near these coastal anthropogenic pressures. And so this might explain why there was more uniform degradation at Plaza and why ultimately it had less coral coverage than Nirvana. So moving over to the social science. So I was asking people if they knew that they were in a marine protected area. And I just want to point out that 100% of tourists had no idea that they were in an MPA. I asked all the stakeholders besides the tourists, do you think that locals know what's going on with the corals in San Andres after we discussed that they are declining? And majority said no, they don't know, or they might have a partial understanding, but very few people thought that the locals have a good understanding of what's happening to the reef around them. So when asked what we can do to help educate locals more about the coral reefs, educating youth really stood out as the main suggested method to help. And then similarly, asked, do tourists know what's happening to the corals? Do they know how sensitive this ecosystem is that they're going to visit and probably dive around? And the majority said no, they don't know, or maybe they have a partial understanding, but nobody said yes, that the tourists know what's going on. So again, how can tourist awareness of coral reef issues be improved? Most people said there really needs to be more education upon entry into the island, or just more signs around the island in general. So I talked to a lot of people. I heard a lot of really interesting things. These three quotes really stand out to me. It really shows that the people who are in the water and who have been in the water for years are really noticing that there's a huge decline of the coral species and that there is a lack of education. One only loves what one knows and one only knows what one learns. So clearly education is really important and it's key. So that was our top recommendation for locals, is really to educate the youth more. So a lot of people said that there needs to be marine science recommended and required in their everyday curriculum at school, and also to have more programs that bring kids into nature with hands-on experiences. And this has proven to be really effective, actually. Studies show that if you educate children about the environment, it increases their ecological knowledge, it deepens their care for nature, and they have more long-term sustainable behaviors as adults. So it's worthwhile. And then it was said that the tourists need to have more of an understanding. If you remember, 100% of the tourists had no idea that they were in an MPA. So what they were saying is that there really needs to be more education upon entry into the island. So when you're arriving, when you're on the plane, you need to fill out a form, or when you're at the island, or when you're in the airport, there should be signs in places, saying you are entering a marine protected area, or in the island itself at the beach. So studies show that it's really important where you put these signs, and so these should be strategically placed, but they can be very effective for education. And so Many stakeholders were feeling that they're doing what they can with the limited resources that they have, but there's also was a lot of frustration towards other stakeholder groups. One fisherman, when he was explaining or how he feels about the government, about Coralina, he said, they say that they work with fishers, but it's just 2 a year. Give more of us a chance, let us help restore the reefs, let us feel important. So clearly there's a shared desire for marine health, but there's a lot of fragmented relationships. And in 2005, when they created the MPA, there was a lot of stakeholder engagement, there was a lot of local support, but since then, a lot of things have changed, naturally, it's been 20 years. National policies have changed, conflicts with industrial fisheries have changed, political tensions with other countries have caused other issues. And the fishermen, a lot of them are telling me, we feel like we aren't at the table anymore. We were there in 2005 when we created this MPA, but it no longer serves us. The way that we fish and where we fish has changed. It needs to reflect that. So there needs to be new zoning consultation and re-engagement with the community. Other challenges, really big challenge is the lack of funding. There's one boat for 65,000 kilometers squared of an MPA. Many stakeholders said that the enforcement is weak and inconsistent. Some called it a paper park. So there's really this struggle to achieve effective implementation just because of low capacity. And so the recommendations are to strengthen this participatory engagement or enforcement by community-based mechanisms, so maybe training the divers, training the fishermen to be your eyes on the ocean, and to improve benefit sharing to reduce conflict. There also needs to be enhanced legitimacy and communication by integrating local knowledge into zoning decisions, conducting regular public outreach, and ensuring transparency in funding, or in enforcement and funding mechanisms. Sorry. And then for the sustainable co-management, the big takeaway here I want to say is the alternative livelihoods. There's many alternative livelihoods for dive operators that maybe aren't doing so sustainably, or fisher operations that aren't so sustainable. And also there needs to be more long-term funding through diverse mechanisms. So to conclude, there was the ecological findings show that there is a decline of these key reef builders to more weedy corals. In Nirvana, it was species specific, where in Plaza it was more widespread. And this might be reducing habitat quality, lower biodiversity, or weakening the long-term resilience. And so the hypothesis of will the disease prevalence negatively correlate with coral cover and cause species composition change, I would say is proven. And the stakeholders, it's really clear that the corals are deeply valued, but there's this change in or non-equitable understanding of the reef. And so there's a large perceived knowledge gap among tourists. So there needs to be more education for the locals, specifically the children, and for the tourists themselves. And management implications, the fisheries feels like they're being excluded from the decision-making. So really there needs to be more co-management that centers local knowledge with alternative livelihoods and sustainable finance mechanisms. So thank you so much to everybody who was on the team with me, and thank you for your attention.
Ready. We start with you, Flo.
Yeah, thanks, Ali, for your presentation. I guess starting on the methods, your photo mosaics for species covers, did you have any challenges in distinguishing between the different species of coral when doing it?
Definitely, yeah. So I'm not really a coral expert. So I was actually, my thesis was on marine spatial planning in the Bahamas until halfway through March. And then the government wasn't able to do research permits and stuff. So I kind of pivoted to this beautiful project I presented to you. So one of my teammates, she kind of did the first month and helped me assign what the species were, because truly I wasn't really aware of all the different species types. I was trying to learn and I did it a bit, but then I would try to do the species and I would send them to her, and then she would kind of correct them for me because it was really expedited. And then I would overlay that onto all the months afterwards and kind of adjust the size of them. But I definitely got a lot of help with the species identification from her, because she is very knowledgeable and I am not.
Okay, great. And regarding the more of the social science side of the methods, what were already the different proportions? You mentioned lots of different groups of stakeholders that you interviewed. What was the different proportion of those groups? Were the samples relatively equal or disproportionate?
definitely a bit disproportionate. There were more, I think there was only like, I think I might have the exact numbers in the annex, but there was, I think, only like 3 or so fishermen, more like 8 tourists. I should say, you can kind of see it here. So dive operators, there was about 6, fishermen about 3, government 5, locals 6 or so, NGO 3, and tourists, I think 8.
Okay, I see. Do you think it was a representative sample of the global kind of view, or I don't know if you can tell given the sample size?
Yeah, definitely huge limitations for sure. I was only on the island to do these for 1 week in the end. So I was happy to get 32 interviews in 1 week. But yeah, definitely there is a bias here. There should probably further studies should show kind of more even distribution between stakeholder groups, certainly, and more. 32 I don't think is a big enough sample size, but it was as many as I could do in the time allotted with pivoting the thesis so late in the game. But yeah, definitely that is a big limitation.
Yeah, fair enough. And were some of the groups easier to approach than others?
So luckily, my supervisor, she introduced me to a lot of some of the people. And so that's why this is here. There might be like a bias for that, for the dive operators especially. But Colombians are so nice, man. They're great. They're really nice. But no, the fishermen were really happy to talk. Those interviews ended up being an hour, whereas the tourist interviews were maybe 10 minutes. People just really wanted to talk and say things, and everyone was really open and communicative. It was honestly probably the more general public, like locals and tourists. I just felt bad being like, hi, do you mind answering a few questions? Like type of thing. But yeah, no, for the most part, I think it was pretty good.
Yeah, okay. Fair enough. Nice. And yeah, I guess coming to the results of maybe, starting more on the social side, you mentioned that it would be important, given the results, to re-engage a lot with the community. How would you suggest going about doing that?
Yeah, definitely. So one of the fishermen actually mentioned there's like going to be a big reggae festival or something on one of the small islands. And he was like, they should, take advantage of when all the communities together. This is our culture. The government or Corlina and the NGOs should, maybe have a booth there and engage people. And I thought that was really a smart idea. Kind of just meet people where they're at. Don't make them go out of their way. But also I think, holding town halls and doing other interviews and such would be a really good way. But then also, kind of just like going into the community itself and really sitting with people. And also maybe through like schools and like NGOs, maybe having like hosting events or something like this, where it could be like a nice event, but then also really trying to understand more and get people involved and really hear their issues. Because at the end of the day, I feel like with marine spatial planning, it's really just a human to human thing. And if you sit down with people and you listen to them and you build trust, and you make a plan that works as best as it can for everybody, then you will have a successful MPA. So you really just have to meet people where they're at.
Yeah, that's a good point. Coming back to you, the first thing you mentioned was the reggae festival. Do you know if that has a lot of locals attending, or is that also bringing in a lot of tourists?
I don't really know for certain, but it was there's different. So I didn't mention this, but it's kind of complex. So on the island, there are like indigenous people who are there. They're Afro-Caribbeans brought over. An English person, long ago found it and then brought slaves over. And so they're not indigenous to the land, but they're Afro-Caribbean, and they're called like the Rizal people, and they're like the traditional people there. And then the Colombians themselves started coming over in the mid 1900s. And the Rizals that I interviewed, they mentioned, the foreigners. And I was like, oh, you mean the tourists? And they're like, no, the Colombians who live here, they're, foreigners. And so this is kind of an issue on the island. But long story short, one of the Rizal people was telling me about the Reggae Fest. So I think it's very Afro-Caribbean vibes. So it gives me the inkling that it's more of a local thing.
Okay, great. Yeah, so in general, kind of considering all these different types of populations, the locals, the pseudo locals, and the tourists, where would you prioritize allocating outreach resources, or is there a way that you think it can be done collectively? What would you suggest?
I think it's always really good to prioritize traditional and indigenous knowledge. They've been there the longest. They know the most about the area. They've seen it firsthand for the longest period of time. They know that the weather's changing based off of the wind. They know about things that other Western people don't really know as well when you're not there. So I would say if you were to prioritize any stakeholder group, it would be the traditional knowledge that's there and to respect that. But I think that all stakeholder groups should be equally, respected, because then it just causes, issues. But if one had to be a bit higher, I would definitely focus on the traditional knowledge.
Okay, great. Christophe, do I have time for one more question?
No, it was screen. Thank you for. No, we're going to.
So thank you,
Thanks,
Alexandra, for this nice presentation. In the continuity of the question, so is there any link between the tourists and the local at some point? In this area?
I mean, yes, because the tourists are really there to. It's 12 kilometers squared, so you're really there to be in the water, So a lot of the locals really depend on the tourists. So, in the restaurants and taking them on snorkeling trips and diving in this kind of way, there is a big link economically, but culturally, I would say no.
So in the economical aspect, they can also implement the cultural and the protection and the awareness of the site. And
Totally.
Okay.
Yeah.
So then, okay, when you mention that there is a lack of education, and regarding your sample size, okay, it's really hard to go straightforward to mention that, and you need to precise it. So where do you think there is a lack of education for you here, and how you will?
Yeah. So I think that the people who live on the island are a bit removed. A lot of them live more up on a hill. I was told that a lot of them don't know how to swim. They live on a 12-kilometer island. So I think that, and they were saying that we know kind of about corals a bit, but we really aren't taught this much. And there's a cultural aspect to it too. Everybody's on a little moped, drinking a beer, and then just throwing the can onto the street. So I think that there is a cultural aspect of that, and that's why they really mentioned educating children, and I kind of agree. I don't think that only educating children is the way, because then we have to wait for children to grow up to then be the change makers, and that takes too much time. But I think educating the children in school, you have math, you have science, and then you should have maybe some environmental education, because they will go home, talk to their family members at the dinner table, they'll educate them more. It can just be kind of the way in more so. So I would say really educating the youth, kind of like they mentioned.
Did you ask them their priority?
Their priority for what?
In the island, for their living. What for their living and a better health of the community? Did you ask them their priority?
So can I have my slides?
Yes.
I hope I put this in here. I don't know if I did. So I asked them this, what is the most important thing about the reef ecosystem to you, if you had to pick of all the things that the reef does? And the majority said in dark blue here is the coastal protection. So a lot of them know that the reef protects them from hurricanes, and they have bad hurricanes.
So they have a good education.
They have
on this.
A bit. So looking at coral knowledge by stakeholder group, I asked them just simply what is your knowledge about coral reefs? And I kind of placed them, because it was a bit of an open-ended question. I kind of put them in 3 different groups: knowledgeable, kind of some knowledge, and then no knowledge. Knowledgeable being I know exactly what a coral is. It has an algal symbiont.
Yeah, but it's a biological knowledge. So if you look at the economical knowledge or the importance for the community survival, they have the knowledge.
They do.
But not for the biological aspect. So Yeah.
Some do.
Unity.
Yeah, some do.
Okay.
Some had the knowledge, like I know that a coral, I don't know exactly what a coral is, but I know that it's good for stopping the wind and the waves and this. And some of them are like, I think it's a rock.
Okay.
Some of them did not know. So it really kind of depended. But again, really small sample size. So, take it with a grain of salt.
Thank you. And how will you link your ecological data with your social data?
I think that, it's hard because with coral reefs specifically, for me, it's kind of, I know, like someone who I was talking to, he's been doing restoration for 8 years, and he was like, you'd have no idea that I've been doing 8 years of restoration if you went diving today. The reef is like really not doing well. So it's kind of hard with coral reefs in that way, but I think that it's just interesting to see that the reef is in need and that people do value it. And so there is kind of this way that I think that you could tell the story of the reef. They don't know exactly what's happening. So if you could educate them, this is what's going on. It's in your backyard. It's happening right now. It will affect you. You depend on fisheries and tourism, and all these things are linked to coral health. If you could tell that story to them, then I think that you would have more stakeholder buy-in, maybe to respect the MPA, because the dive operators and the snorkel groups, they're stepping on the reef, they're picking up starfish and passing it around, and like there's really this.
But why there is all this threat on the reef? What I was surprised, and can you comment on that? It's mainly 2 species in both sides you look at, so that are really decreasing in your length of the experimentation in the survey. So can you comment on that?
Yes.
So yeah. Move on.
So yes.
Come back.
Yeah.
Back. But yeah, this one and the previous one. So there is really mainly just 2 species that are affected. So the light blue, and the other on the other slide is the dark blue one. So the thing is that how you can point. Sorry, I can point on this side.
I'm not really sure about the species themselves. I guess, there are, these big ones are obviously, like these ones, Orbicella is being really widely affected, as it was like the main species in abundance versus it's not in abundance as much anymore. For me, the biggest takeaway is this. This is mortality. These are all species and they're all dying. Like I was diving there, I saw all species. And I was doing the analysis in Tag Lab.
When you say dying, it's affected.
There is mortality.
There is, but okay. How do you access the mortality?
What do you mean by access?
How do you score the mortality? Right?
How do you determine if it's died or not?
It's dying or dying.
Yeah,
Oh, if it's dying or dead.
because if it's bleached, it doesn't mean it's dead.
Yeah, so, okay. Yeah, that's also one of my limitations. So I wasn't able to, if it was bleached, I didn't put it as death or anything like that. So bleached corals aren't mortality, it's just dead corals. And I did the analysis in Tag Lab and sent it to the supervisors because again, I'm not a coral specialist. And they looked at it and they're like, yes, this one's dead. And so they kind of like checked my work. Obviously, I can't tell you if it died because it was bleached and then it died because of the fourth gold bleaching event or because of Skittle D. So that is definitely one of the big limitations. And so that might have exacerbated how much mortality that we're seeing. A dead coral was just a dead coral when I was looking at it in Tag Lab, but there wasn't this intermediary, like almost dead, because the thing with the disease, it's really, the lines are really like exact. So half of it could be dead and the other half would not be dead. So only the half that was dead. And the other half of the coral was still alive, even if it was a coral this big. So I tried to be really specific in, what was mortality and what was probably soon to be mortality. But if it wasn't dead yet, it was not dead. If that makes sense.
Okay.
I don't know.
I'm fine.
Yeah, we can go back to you, Aziz, with a couple of questions.
I have a quick question on the fishermen, because I don't really care about fishermen. So you said 20 years ago, sorry. So 20 years ago, they helped to create this MPA.
Yes.
What are they unhappy about now that they think they can implement? What changes do they need?
So they've told me they have to fish deeper
find fish now. So they're not fishing in the same places that they were fishing before. So spatially, the marine protected area, the marine spatial plan needs to change. The spatial part of it is not congruent anymore. Where they were fishing, where there was fish in 2005, there are not fish anymore. And I think in the zonation, that probably needs to be reflected of that, with some no-take zones or still allowing them to fish in certain areas. Also, the indigenous people, the Rizal people, get more fishing rights than the Colombians do. That's really pissing them off, to be frank. So there's this cultural shift. And then also Nicaragua has some access to some of the area for industrial fishing. And that is causing a big cultural shift, kind of like, well, if they're allowed to do it, then I'm going to do it too. So what's the point? So there's a big kind of cultural, intergovernmental divide.
And just going off Aldine's point of the disease to mortality on slide 20, you used photomosaics. Is this photogrammetry? You use photogrammetry for this, or is it just purely for the photo, for tagline? Or were you building a 3D model?
No.
No, okay. Can you just explain how, when you put this image into TagLab, what are you actually doing in TagLab? Can you just briefly?
So I am kind of tracing the coral itself, and then it fills in the area, assigning it with a species with help.
AI?
No. Oh, I think that they do use AI. Yes, they have the program that they've done all of that. I'm just like given the TagLab, but yes, they have a model that they're doing, which I don't ask me about, respectfully. And yeah, so I'm basically tracing whatever is. So if this was dead and these little parts were still alive, then I would trace those little parts that's still alive, and the rest of it would be dead. And then so this whole entire thing would be all colors of coral species over the actual image. And I take those little basically like blobs of coral as a shape file and put that and lay it on top of 2 months later actual photo. And then I edit if the coral grew, which it probably didn't, or if it shrank, or if it was completely dead and covered in algae, then it was deleted because it died. And then those changes are all exported into the shape files, are changed into CSVs. And so then I can track the difference in area between each month.
So you did this with 3,000 colonies?
Yeah.
How long did that take you?
Very long.
One final question. What's the problem with weedy species? Because towards the end, these weedy species, they allow the reef to persist.
Yeah.
They're usually reproductively reliable. So if they're not reef building species, what are they?
I think that, to me, as a non-coral expert, the issue would just be that there is obviously a big lack of the biodiversity that was there in the first place. And just by being there and looking at them, the reef builder species were much larger. They could probably house a lot more fish and have maybe more ecosystem services along with them in their size and all of that. And then these weedy species are much smaller. And so I think just on total coral coverage level, there's just like huge gaps. And you're seeing, it's just like it's like a graveyard essentially. And some of these little weedy species pop up. Maybe over time they will replenish. One person actually told me, it was the only like good remark. He was like, yes, they're dying, but I see little corals growing on top of dead corals. Like these things happen, and I'm hopeful, and maybe it will be resilient, and these weedy corals might pave the way towards such resilience. The issue is that Stoney coral chase loss disease is in the water, and they don't know how to get it out of the water, and so once it's there, it kind of persists. But yeah, maybe the weedy corals could be the key to the resilience of the ecosystem. It's possible.
Okay, thanks. So we have time for 2 questions, and one is already there. Thank you, Andrea. So I find this question interesting. Were questions done in English or Spanish? And so
yeah, so with the Rizal people, they speak English. They are actually really upset that the people are coming and making them speak Spanish, and they're losing their language. So they are Afro-Caribbean and they speak English. So a lot of these interviews were in English. The tourists are tourists, and therefore a lot of them spoke English as well. And then I had my, I've been living in Barcelona and taking Spanish classes. And also I had my interview questions in Spanish, and I kindly asked if I could record them. And so if the person, was more comfortable in Spanish, and I told them, if you can answer a better answer in Spanish, please do so. And some interviews were done in Spanish if that's what the participant needed.
And we'll finish with Nicole.
So kind of putting together all the things that you did, right? You noticed that stony coral tissue loss disease is a big problem, but that also you want to incorporate different locals. So my question is, what is being done now to control the disease?
Yeah.
And how can you incorporate people to those kind of measures as well?
Definitely. So they made an antibiotic. I think in Florida, or a conglomerate of countries, because like I said, this appeared in Florida in 2014. So it's been 11 years that we've been dealing with stony coral tissue loss disease. There is an antibiotic. It looks like this. It's kind of like a paste that you put. And like I said, there's really definitive lines of the disease. And so you can put it on where the disease is affecting it. This is the healthy coral. This is the disease. And it will manage the spread of the disease. The thing is, it has to be done multiple times over and over to make sure that it's working. 65,000 kilometers squared on San Andres, I think, including the NGO I was working for and the other NGO, maybe there's five people who are certified to do that. It's just not enough. And so, yeah, would be a great alternative livelihood for dive operators, or these tourist type of conglomerates that are doing not very sustainable tourism, or fishers or stuff like that. You want to keep them in their same livelihood. They like being on the water. They like being out on the boat. They don't want to sit behind a desk. You can't tell a fisherman to go do a desk job, but maybe you could train a fisherman to dose antibiotics on the reef, and that could be a way that they have stable livelihood and protect the reef at the same time. So, yeah.
Thank you. Congratulations.
Thank you.
Lila and Gaia and Fabian and Andrea saying also congrats, not only asking questions. So we're good. We're coming back here at 11. We're starting again, so please be there just before 11. We are now taking a break. So there is breakfast available on this floor terrace. Okay. So enjoy, discuss, coming back here just after 11. On y va. Are you ready? Yes. Hi. So here we're back. Florence, good to have you again. So we'll continue now with Cloé, and Luna, center. Cloé, you're ready? We'll just share your slides. You should be able to change the slides directly. Can you just check?
Yeah.
Box? Yes. Okay. Ready? Very good.
Okay. Hello, everyone. So today I'm going to present you my internships that I did in the Philippines, which was about the deployment of the Sulu Marine Corridor, and more specifically on the ecological and methodological baselines to assess the effect of artificial structures on habitat complexity and connectivity. So let's start by a bit of intro. So coastal communities in developing countries are facing combined pressure about climate change, increasing demand for marine resources, and the limited regulation and enforcement. So this leads to an overexploitation of the stock of fish that are already depleted. And this ultimately threatens fisheries, biodiversity, and food security. Just for a few numbers, in Shark Fin Bay, so where this internship has been done, it's up to 94 of total catch from artisanal fish fishing that are reef-associated species. 83% of the people in the bay consider fishing as vital for livelihoods. 17% observe a decline in fish resources, and 73% identify destructive fishing as a major issue. So what can we do to protect those marine resources? We can do marine protected area. So it is well known that when it's well managed, it increases diversity, abundance, and size of the fish. But what can we do to complement this passive restoration strategy? We can do active restoration. It's defined by the process of assisting the recovery of an ecosystem that has been degraded, damaged, or destroyed. So one of the tools of active restoration are artificial reefs. They are known to increase connectivity by acting as stepping stones between natural and artificial habitats, and the fish are regularly seen moving between the two. So I introduce now the concept of ecological corridor. So on terrestrial ecosystem, they are narrow habitat strips that connect isolated patches. They facilitate species movement, they enhance gene flow, they support recolonization, and they also increase biodiversity. In marine environment, not much has been done concerning ecological corridor. So what if, rather than relying solely on the creation of new reserves, we could reconfigure existing ones to enhance density and connectivity within patch reefs? So in this experiment, we have two sites. The garden, which is a part of the fringing reef of Cayman Islands, and the pinnacle, which is situated 200 meters away. And the pinnacle has been affected by past dynamite fishing. So here on this picture, you can see the garden, which is very rich. It's actually the most productive part of the fringing reef of Cayman, and the pinnacle. So this is the results of the dynamite fishing, and it has active restoration has been carried out, especially for coral restoration. But even with that, the fish population struggle to come back. So the idea here is to assess whether an artificial corridor can support the recovery and the resilience of a degraded reef, here the pinnacle, by connecting it to a more productive site, here the garden. So I've been working on 4 different research questions. The first one was the comparison of ecological indicators between the garden and the pinnacle. Do they support the need for a corridor? The second one was, can artificial structures create new habitats and influence the fish assemblage on this muddy substrate? The third one was, how do the structure affect the animals that were already present here in this environment? And the fourth one was the early patterns of colonization on the structures to document the colonization. So let's jump to material and method. So Cayman Island is a small private island of about 4 hectares situated in Shark Fin Bay, northeast of Palawan, but southwest of the Philippines. The study area is within the first marine protected area in between the 2 reefs previously mentioned, on a flat, muddy bottom at the end of each reef slope. So we have 1 control site and 1 corridor site. And the corridor site is made of 17 pyramidal shaped structure, as you can see here on this picture. This is 1 unit, and they are all aligned after each other on the 200 meters distance. They are in depth between 17 and 24 meters. And on each unit, we have added some biohuts filled with shells to increase microhabitat complexity and offer protection for the fish. So to assess the impact of the corridor, I've been following a passive design, which is a method that compares the changes in an environment before and after an intervention on both an impact site, so here the corridor, and a control site. To collect my data, I've been using 2 underwater visual census methods. The first one was fixed transect along the 2 sites. So here is the control site, here is the corridor site. Here in green is the 50-meter transect. And the second method was fixed point on 3 different structures to assess the colonization, and this was done with quadrat-like method. So each research question have specific protocol. So for the first one, the comparison of garden and pinnacle, the targeted species were only commercial fish. The second one, the creation of new habitats and the influence in fish assemblage, it was any pelagic fish that I could see on my sites. And for the how does it affect the composition and abundance of the animals that were already present on the site, I've been targeted all the mud-associated organisms, so all the benthic fish and invertebrates. And for the colonization, it was interesting and solitary organism, but only seen on the surface of the structure. I've been working with different variables, so species richness, abundance, size, biomass, age category, and percentage cover for the colonization. The age category, it was either adult or juvenile, and it was estimated based on the size and the sexual maturity of the fish, depending on the species. So I will be presenting results and discussion in the same time for each research question. So the first one, the differences between the garden and the pinnacle. So I've been analyzing the Sulu Bay dataset from 2021 to 2025 because they do annual survey in the bay. So what we can see between the two sites, firstly, is that there is no difference in species richness. So it's the same number, approximately the same number of species that we see on both sides. But there is an abundance, there is a difference, in abundance, with the garden being more abundant than the pinnacle, and also for the mean size of the fish, with the garden having bigger fish. But the most relevant indicator was the biomass, which is defined as the total mass of living organism in a given area expressed in kilogram per unit area. Here it was in hectare. So we can see here that the biomass is really higher in the garden than in the pinnacle. And what is interesting is to see the trends over time. So for the garden, it keeps increasing over time. And for the pinnacle, it tends to decrease. Even if the slope is not very big, it tends to decrease. So what we can say is that species richness and diversity are comparable between the two sites. So the risk of ecological imbalance after the intervention is quite low. The garden is more productive, even there is a higher biomass compared to other MPA in the Philippines. So connecting the two through a corridor could actually support the recovery at Pinnacle. And knowing that connectivity is essential to promote recolonization and enhance resilience in degraded systems. So this configuration could act like a source-sink dynamic with the garden being the source. However, we need to be careful because there was a high variability in the dataset that I have analyzed across the transect and through the years. This leads to a low statistical power. So we need more frequent monitoring and over the long term on those two sites in the future. So the second part was the creation of new habitat on the muddy substrate, how it influenced fish assemblage, and how it also influenced the fauna that was already present on the sites. So what we can see is that before the intervention, we had 187 observations from 16 different families. The first family dominating the assemblage was the goby, clearly, from far, followed by the Lujanidae, the Signatidae. After the intervention, the abundance jumped on to more than 2,000 observations from 26 different families. And the dominating was the Casonidae, followed by fish that couldn't be identified due to field conditions, Lujanidae, Apogonidae, and goby only at the 5th position now. And we can see that it's mostly on the impact site, no more on the control. So on the impact site, we had 18 new species of fish appearing that were never seen before. 5 of them were described as mud-associated, 8 reef-associated, and 5 ecologically or commercially important. So about the richness and the abundance of those two sites, so what we can observe is that there was no difference between impact and control sites before the intervention, no difference before or after the intervention on the control site only, but the impact sites show significantly higher species richness and abundance after the installation of the structure. So more specifically, in average, it passes from 11 to 27 species seen, and from 35 individual to more than 600. This peak in abundance was mostly dominated by juvenile fish. So we had more than 500% more in abundance in juvenile fish. So, but on the other hand, we had no difference on the control site, either over time or between adult and juvenile. And we also had no difference in adult, both on the impact and the control. So it's really the juveniles that have exploded. One of the research questions was to know if there was an impact on the fauna that was already present on the sites. So no significant difference has been observed for now. So this suggests that these communities remain stable even after the implementation of the structure. So this increase in species richness and abundance is not very surprising because we know that artificial reef increase habitat carrying capacity and tends to enhance those indicators. The recruitment of juveniles is not really surprising either, but even if it was not anticipated. But there is like three main factors. It's like the natural nursery role of muddy habitats, also the known attraction of juvenile for artificial reef, and the specific design of biohuts that are made to attract juveniles. So interestingly, Eugenus vita and species from the Casio family were present on both sites before the installation of the structure, but only present on the impact site after. They were not seen on the control anymore. So we can just feed the debate on whether artificial reef enhance fish production or just redistribute existing populations. So once again, we need to be careful because artificial reef and an increase in connectivity pose risks. So it can modify sediment composition, increase organic matter accumulation, facilitate the spread of invasive species, and attract predators. Here in these studies, the conclusions are limited by the small number of replicates and also the short post-deployment monitoring period. So even if the first results are promising, we still need more long-term monitoring to detect any potential negative impact. The last question was about the early patterns of colonization on the structures. So we can see that it just followed the classical temporal sequence, beginning with biofilm, followed by algae and cecine invertebrates. And here we had a very strong positive relationship between immersion time and mean coverage, with the turf algae largely dominating the colonization. We had also some acidian and bryozoans that kept increasing with time. And from the 43rd day, we also had hydrozoan appearing. So now we are just waiting for the structure to attract higher trophic levels and get to a climax community, a more stable community. So just to get back to the research question quickly, so does the comparison of ecological indicators between Garden and Pinnacle support the need for the implementation of a corridor? We saw that, yes, Garden is more productive than Pinnacle, whose low biomass support the need for the corridor. Can artificial structures create new habitats and influence the fish assemblage? We saw also that, yes, rapid and significant increase in species richness and abundance was recorded, and mostly with juvenile. The third one, how does the structure affect the animals that were already here? We saw that for now, there is no shift in communities that has been observed. And the early pattern of colonization just follow expected pattern in the literature. So in conclusion, while adult fish movement and connectivity are yet to be confirmed, the early results suggest a potential for the corridor to be tested, refined, and replicated elsewhere in the bay and beyond. Thank you.
It's not over.
No. The worst is yet to come.
Allez, let's go for Adine.
Thank you, Cloé, for this amazing work. And I really like this idea of combining this NPU with the reshaping of the ocean floor. I mean, geological time set up a specific geography of the site. I think that maybe now it's time also to help implementing some paradise area for new fish population and other animals. So it's really interesting and insightful. So maybe I've missed some detail, but when during the day did you access the biomass and the abundance and so on? So when have you done the transect and which season?
So the biomass, how I calculated it, it was with the data set of Sulu Bay, and Sulu Bay is doing an annual survey every year in May, April, May. And so this is done on 4 different replicates, 4 different transects, a bit everywhere in the bay, and on those 2 sites.
Okay, so it's only once a year.
It's only once a year, yeah. Yeah, that's why I mentioned that it's a bit of a limitation, because at the end my statistical power was a bit low.
Okay. Really, really nice. So now that you have all this expertise in the topic, and so what are the limitations of such approach? And do you think it's sustainable on long term?
So the limitation maybe it's can be the difficulty of collecting the data over the long term because it's a project that it's meant to stay a long time the structures are made with like concrete that's gonna last a very long time in the water. We calculated it's like maybe 80 or maybe 800 years old. So maybe the difficulty would be to find people over a long time period to like keep collecting data on the project. Also, if it has to be replicated in the bay, we will need to find like some arrangement with local communities to make sure that the corridor is not affecting the fishing, because now it's in the marine protected area, so no fishing is allowed. But then if we want to replicate it outside the marine protected area, we will need to discuss with all the stakeholders to make sure that everything is done correctly and everything like it's fitting everyone activity. So this can also be a limitation.
And did you start to exchange with the local community, with the fishermen regarding all this implementation on the side?
Not yet, because it's really the start of the corridor. Like during my internship, we installed the structure in the water. So I had like not even 1 month of collecting the data after the deployment of the structure. So now before starting to discuss with everyone, we're just waiting to see if it's worth it, see if it's working before starting to discuss.
So then before setting up such structure in the natural area, so did you have to ask any permit or authorization from local community or from the government?
Yeah, so there is permit to ask for. As it's within the first marine protected area of Pangatalan, we had some derogation to put the structure. But if it's going to be outside the marine protected area, it's going to take maybe much longer and much administrative stuff we need.
Okay. And how did you give feedback, or did you give any feedback on the local population regarding your project and the positive impact of such implementation?
So right now the corridor is really a pilot experiment. So we haven't been talking about it like outside Sulu Bay and outside the scientific team and everyone. For the creation of MPA or this kind of project, of course, yes, like public hearings are on place, like we ask for the communities their advice and like we just work together. But for this specific project, it hasn't been just not yet. But if it's gonna work in the future, yes, of course, we will do some communication with everyone.
Okay, thank you so much.
Thanks again. Thesis?
Thank you very much. It was really interesting. How long has it been there? Sorry, maybe I didn't catch it in the presentation. When was it implemented, the corridor, and how long?
So I have access.
Sorry, yes.
Timeline. So basically, first, when I arrived, it took a while for the literature review, for the method and the protocol to be designed, because it was a big part of the work to think about how we're going to implement this PASIPS methodology. Before, after, control, impact, we needed to like install the site even for when the structure were not there, just before. So like it took a while to install just the site. After I had the data collection phase, but from the before installation phase, and then the deployment of the structure took approximately more than 3 weeks. And then I had another small month to collect my data for the after phase. So that was, and in the meantime also, when I was doing my data collection for the before phase, the construction of the structures were ongoing. So we had to deal with that too at the same time.
I like the idea of it.
But
There's major ethical considerations, which I actually don't mind, but there is, like, and you mentioned them in your limitations about the hydrodynamics. So over this timescale, you have no idea really how you've affected it. Do you think your results are the consequence of you perturbing an environment? So for example, there's papers already from Thailand, from Taiwan, Japan about kind of a big boom in juveniles after you create an artificial reef and then everything settles out. Do you think that's what you're seeing?
So yeah, for now you're right. It's way too soon to conclude any strong effect. But I think it's really a matter of contextualizing the project, because here is just 200 meters in a 6,000 hectare bay, which is mostly dominated by muddy substrate, coral reef, but muddy substrate is largely dominating the area. And so the site that we are connecting, it's degraded. It's just not a normal site. And so if we don't do any active restoration, we might just get this site just degrade more and more. And it's within the first marine protected area. So instead of just looking without doing anything, it's still better to try something and see with time, because the project will be followed scientifically. It's not just some things that we put bet and we see. So if we see that there is potential negative impact or anything, we will adjust, and the structure are reversible. We can still take them out. But yeah.
That's my other question about the pinnacle itself. Why hasn't the restoration worked? Like, why you mentioned there was some restoration on there. Like, why?
Maybe because there is no connectivity, and so we are just
for 200 meters.
Yeah,
No
I know, and larval connectivity, probably it can last for thousands of kilometers, but adult connectivity with big fish, sometimes they just need help to because the muddy substrate is quite hostile. They don't want to go, they don't cross by themselves because of the predators, so we're just thinking helping a little bit this natural dispersion with the corridors that could enhance the restoration on the pinnacle.
On slide 16 and 17, maybe I've misunderstood. Sorry, the slide 16 is before.
Yeah.
And slide 17 is after.
Yeah.
So there is a change in composition.
Yeah.
But in your conclusion, you said there's no change in composition.
So there is no change in species richness and diversity. So I've got some. Okay. Here it's the. Oh wait, no, I get confused. So yeah, there is a change. No, I was saying in my conclusion, that between garden and pinnacles, there was difference in community structure.
Okay,
sorry.
That's my mistake, sorry. Okay.
France?
Yeah, thanks, Chloe, for your presentation. I also had the pleasure of reading your report as well, which, it's great, innovative project. One of my questions, I think, is to follow up on, you spoke to Aldine about the limitations in monitoring. I'm curious about the limitations in the methods itself, the use of artificial reefs. Do you have any limitations to share regarding that method?
You mean the method to collect the data on the artificial reef?
No, I mean using artificial reefs themselves. For regarding complexity, for example, of artificial reefs.
Oh, sorry, it's just the connection.
The creation of.
The creation.
Yeah, sorry. Just one. Yeah, exactly.
Well, we know that it can, it's a fact, it can affect the substrate, it can affect the community, attract the predators, and have some impacts that we didn't really anticipate. But in this kind of configuration, once again, I think it's better to try some things than to just don't do anything. And the potential effect on the fauna that was initially there is being monitored, and it's going to keep being monitored for the rest of the life of the artificial reef. And so right now, the results say that there is, once again, it's a very, very short post-deployment period, but it doesn't have any significant effect on the benthic fish and the invertebrates that were already there. And I also saw that on the literature, sometimes artificial reef can even attract invertebrates and make those communities more abundant. And so invertebrate being like very low in the trophic chain, that will like all the ecosystem will take place. And actually, I don't know, it's not going to answer to your question, but this project have been inspired by a pipe which is linking Pangatalan Island and Dipla Island, which is 400 meters away. And this pipe is made of concrete blocks to maintain it. And 15 years after, we realized like a full ecosystem have been created. There is sponges, there is corals, there is fish. So we wanted a bit like to reproduce this to like linked to ecosystem. So I think we might get more benefits than disadvantage. And also knowing that the muddy substrates are quite poor in terms of biodiversity compared to coral reef, mangrove, seagrass, or it's very low. So yeah, it's better to try something.
Yeah, those are great points. And I think what's key to keep in mind as well when you're creating artificial reefs is that they have sufficient complexity to attract both juveniles and adults. Even, I mean, attracting adults is more tricky, but do you have any ideas of how the structures you put in place could be improved potentially to attract adults as well as juveniles?
Yeah. So first, we didn't want it to like put some corals on the structure or anything. We wanted the natural colonization coming by itself. Don't interact too much with the habitat. Then after we added the biohut, so it's one thing that we added on the structure to like increase the complexity of the habitat. So that's one thing that is surely acting for the development of the juveniles and everything, because it's made to attract juveniles. Then how we could improve them, I guess the job will be done by itself naturally, like when the colonization will come and when the sponge will start to come and the corals will start also. We had some evidence on the concrete blocks that take the site together. There is already coral appearing on those concrete blocks, and it's like not even 3 months after their immersion. So we're gonna just leave the nature do its job by itself and see how it ends up.
Great. And how did you choose those structures? Whose idea was it to get those structures, and where did it come from?
So both from the pipe that I've just mentioned before. So the line, the structural line after each other was from the pipe, and the pyramidal shaped was also another inspiration from. Because Sulu Bay have been doing active restoration for the corals with some other artificial structures. And at some point in the bay, it does like some little mounds, a bit in pyramidal shaped. And we saw that it attracts a lot of fish, and the abundance of fish was really high on the structures. So it inspired like the pyramidal shaped and the line, the pipe. But also in the literature, it's a shape that it's currently done for artificial reef, to put it just in triangle like this. So it has been done also. Okay,
great. And you spoke about the possibility of replicating this corridor elsewhere. What would be your key takeaways or your key tips for implementing it elsewhere? What would you do potentially differently?
So the first thing, I think it could be very interesting to study the connectivity before installing the structure. So genetically speaking. So like for here, for example, before installing the corridor, it could have been very precious information to know if the two populations on the garden and the pinnacle were actually the same, or were a subpopulation, or what was like the genetic similarity between the two populations before connecting them. Because like this, when we do after a while, after a few years, when we will do genetic analysis again, we will be able to see if genetically the population changed. So this is what I would suggest to do for a potential replication, like the biggest thing that I would suggest. After, maybe also try some different shape. of artificial structure. Because I also saw in the literature that the triangle shape is widely used, but it's not the most efficient. So maybe try some other shape which works better. But yeah, that's mostly it.
Great, thanks. And just a last question, maybe, how would you...
Sorry, so Florence, I have to cut you just because I would like to pose the questions here also if you do have questions. And I'm sure you have. Yes.
Did you, for the material used, the concrete, did you use like different viscosity or porosity of the concrete, or is like one for all the?
So it's
She can just repeat the question.
so what is the concrete that has been used, and did we look at the porosity and everything? So the concrete that has been used is the same that has been used for the previous artificial structures. It's a ternary blend, something. It's made like from west, actually like half west and half cement. So it's kind of eco-friendly thing, if we can say that. But it's meant to last a long time in the water, in marine environment. That's all I can say about the concrete.
Is it smooth on the surface?
Yeah, it's smooth.
You take your question.
Mine just felt like it's half wet and half concrete.
Half wet, like the waste
Okay.
of previous cement and
okay.
mixed with
Recycled.
yeah, recycled and yeah.
My question was, so you said, of course, you have some good initial results, but of course, it's very short time, so you need to wait a long time for the impact. So, but there's also maybe an idea of replicating it elsewhere. How long do you think you would have to wait? If this is the test, like how long do you think before we say, okay, it was a good idea, let's do it again?
Okay, so we targeted 2 to 3 years. 2 to 3 years monitoring to potentially see, like, to have a really good basis to see, do we replicate or should we just leave it like this? 2 to 3 years. But now there is someone that is taking over my project, and it's going to be probably the same on the next year. So the monitoring will like keep going. So we will have a big data base. So it's
2 to 3 years. It's based on what, like hope or?
Yeah, a little bit of hope and brainstorming, I guess. No, yeah, it's it was the correct time for us in our mind to say like, okay, after 2 or 3 years we're gonna start to have like concrete results. And
That's a growth of fish before maturity or anything?
no, we just discussed about it like together.
Okay. Last question? Are we good? Yeah, of course.
Wow.
Does this shape, like this corridor, does it exist in nature? Because I was just wondering, just from a self-organization perspective, if this like small corridor is sustainable in the long term, or would you have to keep using like these activities?
If this shape is
Yeah,
done
if
in
it just
nature.
exists in nature, it is very thin line of reef.
So actually I dove one time next to Pangatalan, and my dive reminded me the corridor in the shape that it was made. So it was really like some kind of big peak going out of the substrate and just a line after each other. So I was even surprised. I was like, oh wow, that's a natural corridor. But I'm not sure, like really like this, if it's in nature, but can be similar, yeah. And for now, so this has not been documented like in this study, but we already see some adult fish crossing the corridor, and some big ones that appeared and were not here at the beginning, like the groupers, or so it's already starting to have some kind of connectivity. So it's really like we have hope. But yeah, we still need to know how we're gonna analyze this connectivity, how we're gonna manage everything. But big fish are already there. So.
Thank you, Chloe. Congratulations. You've got also some support initially. Hassan is still there. And and yeah.
Thank you, guys.
So congratulations. It's customary, so I'm if you want to say anything, like you cannot ask questions, and none of what you can say will be considered in a grading or anything. But as you travel, if you want to comment or anything.
We received good question from you and from this guy also. The idea is to understand what is happening in the high, because you have different level of biodiversity. And if I can explain more the last question, all the in the past we tried to experiment something like that, not really like that, but something. After 5 years, I count 120 big fish, almost 2 kilo, the minimum, and the rate is 2 kilo by structure, from each structure. And it's for that we need to do that. And the connectivity is really important, and Cloé answered at the end. Just one more, 3 weeks after she left, the huge fish stayed, and when we approach the first structure, the fish go on the second structure. It's never get out. He follow, and sure, the result will be perfect.
Just one thing.
I think we can hopefully continue this discussion during lunch. Yes, but I think it's time for Luna now to go ahead. So just checking it works. That's good. Yes. Okay.
Yeah.
So with Luna, we're in Philippines, and I guess we're going to talk a bit about Cayman Islands.
About the Cayman Islands.
Yes.
Ready?
We are.
All right, hi everybody. My name is Luna, and today I'm going to tell you about my internship that I did in the Central Caribbean Marine Institute, where I explored the impact of video on ocean education and public engagement. So you're probably all familiar with this picture. It comes from the latest documentary with David Attenborough, and I want to ask you a question about that. What if you could ask your question to Sir David? What if he could answer you in real time? How could that change the way we learn about our oceans? So, he said it himself, that after 100 years on this planet, almost 100 years, he now understands that the most important place on Earth is not land, but at sea. And it's true. It covers 70% of the, of the planet, and it regulates the climate, it sustains biodiversity, and it sustains billions of people. But yet, the public understanding is still very limited. There's a lot of pressures on the ocean, and we need to have effective marine conservation to make it work, and that depends on regulation, but also public awareness and education. So, those regulations kind of started in 1972, especially with the UN conference in Stockholm, where the first global frameworks for ocean protection started. But also a bit later in 2005, the concept of ocean literacy came into place, which is basically understanding the ocean's influence on you, but also how you influence the ocean. And it comes with 7 principles to guide education. However, today, marine topics still remain underrepresented in school curriculums. Actually, 10% of the children feel they know about solutions about climate change in the US. And we need to find a way to bridge that gap. And a way to do this, we need accessible, engaging, and emotionally resonant tools. This could be done with multimedia learning. So traditional education struggles to make ocean science accessible and also memorable. We struggle to make a connection with it. And what we could do is use visual media to actually simplify complex topics and strengthen emotional impact, especially for youth. And we could use interactive live broadcasts to foster stronger connection with these people. And that's why Reefs Go Live comes into place. It was created by the CCMI, and it's an underwater live broadcast where divers go underwater wearing full face masks and teach a full science lesson to the people watching. And this broadcast is aimed at year 5 and 6, so it's about 10 years old for the children, but it's also for everybody to watch. So we are connected to the internet and everything is live, and people can ask questions, and there is a conversation between a topside host on the boat and an underwater host. We also have cameras to illustrate what we're saying. With that, I wanted to look at how effective are different video-based communication formats in promoting marine science education, engagement, and conservation action. And I expected to see that live, interactive videos will lead to better learning, engagement, and conservation motivation. So my study objectives really were to study the effectiveness of knowledge retention, emotional engagement, and conservation behavior. Underwater, and then compare, sorry about that, it didn't look like this before, but anyways. And then to compare two formats of videos, underwater live broadcast versus a pre-recorded documentary. And also this study was to learn about how to make it better, because this is something completely new for me. So I wanted to learn how to make it, how to do it, but also how to improve the reefs go live to make it even better. So for the methodology, the location was on Little Cayman in the Cayman Islands, and the study format was 4 episodes of Reefs Go Live and 1 pre-recorded video documentary. So for the 4 episodes, the first one was on seamounts, and it was quite educational. The second one was on cryptic creatures, and it had more of a light tone to it. The third one was on coral restoration, and it was more action-based. And the last one was on climate change, and it was a bit more emotionally driven. The pre-recorded video was a short video, about 7 minutes, and it was about the Bloody Bay Wall. Bloody Bay Wall is a marine protected area on Little Cayman, and it relates quite well to the Seamount episode because it looks at how topography is really important for marine ecosystems. And it uses edited videos and expert narrations, doesn't have any interactivity. For the participants, it was obviously the students, year 5 and 6, and the general public, and they were all recruited via schools, CCMI, and online platforms. And for the video, I recruited the participants through the broadcast surveys. So I asked them a question if they wanted to participate in another experiment. For the surveys, it was a pre and post survey, so before watching the broadcast and then after watching the broadcast. And they were all digital, anonymous, and the consent was asked every time. We measured the knowledge, the emotion, and the actions. So for the results, we had a total registration number of 413 people. So that's the total number of people that registered to watch a broadcast. But actually 250 virtually attended to all the broadcasts. Well, it's a total number. And for the last episode, we had an event at the Grand Cayman Cinema where people could actually watch the broadcast, and 560 people attended to this. For my surveys, I had 364 people answering my surveys total for the whole season. Most participants in the broadcast and in the surveys came from the Cayman Islands, and then the US, and then France. I wonder why. And the age ranged from 10 years old to 80 years old. And before looking at the knowledge retention, I wanted to look at the baseline knowledge of people and see if it was related to age and to also the location. So I had 3 knowledge questions in a pre-broadcast survey that looked at the total knowledge score. And basically, I found that children had the lowest score compared to adults. And it was significantly different. Apart from that, between adults, the age groups weren't that different. And I was curious to see if coastal proximity was also influencing the baseline knowledge. And it turns out that people living near the coast scored a higher knowledge score before watching the broadcast than the people not living near the coast. Now to look at the knowledge retention after watching the broadcast. So I just want to remind you that the surveys were anonymous, and therefore it was difficult to look at the actual individual's knowledge retention, So I asked the question to see if they completed the pre-survey before doing the post-survey. And 83% of them say they did, and I thought that was strong enough to make a comparison. For the episode, there was, episode 1, 2, and 4, there was a significant difference in knowledge retention. They all scored higher. But from episode 3, they seemed to score a bit less. Well, not less, but it was not significant. They seemed to just know about corals. For the emotions that all the episodes elicited, most of the episode 1, episode 2, and episode 3 brought the feelings of inspired and hopeful, curious, and engaged, and the last episode brought a bit more concern and worriness. Now, to look at the action, I had a list of action for people to choose from and to say how frequently they do. The light blue is post, and the dark blue is, no, the light blue is a pre, and the dark blue is a post. And basically, the common actions remain stable. But what's interesting to see there is that less familiar ones increased a bit more. So advocating for the protection of marine habitats and looking at using sustainable seafood, eating. Anyways, for the pre-recorded video, what I want to mention there is that I had a really, really small sample size for the video, but 100% of them watched the video and filled the pre-broadcast survey. Pre-video survey, because that one was not a broadcast. And then I looked at the difference between the episode 1 on seamount of knowledge retention and the documentary. So I chose episode 1 because of the similarity in the topics. And it turns out that because the people watching the video scored 100%, there was a significant difference in the knowledge score after watching the video. For the emotions, the live broadcast, people felt a bit more curious and engaged with this episode, while for the video, there was a broader mix of feelings. So there was a bit more of everything, but also a bit more of people feeling concerned and worried. Now, to discuss about all this, we found that the baseline knowledge of marine science was higher in older people than in young people. And people living near the coast also seemed to know a bit more. And that is supported by a study that was done by the Back to Blue Initiative, where they asked about 3,000 young people. And they asked about marine science and marine topics. And it revealed gaps in ocean literacy. Actually, 50% of them, young people, do not understand how ocean impact them, and vice versa. 47 of them think the ocean is healthy, and only 28% believe that they are individually responsible for the protection of the ocean. More than half of young people now turn to media and film for ocean information, and only 1 over 3 use formal education to learn about those things. This really highlights a gap in marine science coverage in school. Now, we also found that there was an overall knowledge improvement after watching both the broadcast and the videos. And this makes sense because today's students are digital natives. They are always on their phone. They're always watching videos. So it does make sense. Traditional learning doesn't work anymore. We need to try and find a way to make them understand for real. And having a real-time Q&A can clarify complex topics and deepens understanding. And actually, there was another study that showed that there was a 25% improvement for students that use interactive live video. We need to find a way to make engagement and emotional resonance a larger role in all these learning methods to make the outcome better. Now, the live broadcast captured direct attention, while the documentary led to a bit more reflection because of the wider mix of emotion. And this is interesting because we could combine both formats together to actually make it work and maximize learning. What we could do is have this pre-recorded video before watching the broadcast, make the people understand what they're gonna learn about, and therefore they'll be able to connect more during the broadcast. This also shows what we found with the emotions, the importance of framing theory, how effective communication strategies balance optimism with urgency with optimism, and obviously present the issues with hope to prevent audience discouragement. Now for the behaviors, routine actions like recycling, they remain stable, like I mentioned, and there were more shifts in less familiar actions. And there was another study on that people watch climate documentaries, and it showed that it sparked immediate motivation to act, although these faded over time. Well, some stayed, I guess, and some didn't. But it really supports the fact that our educational media that we did with CCMI reinforced existing habits and also nudge viewers towards new behaviors. Now, there was a lot of limitations to this study. The first one, and I'm really happy to talk more about them after, but the first one really is the fact that they were anonymous. So we cannot look at individuals' knowledge retention and actually assess the trends. The self-reported data may be biased by social desirability or recall error. The unequal sample size and also the sample size themselves, they were quite small for each survey. And the short-term measurements, it would be interesting to look at the long-term knowledge retention. No control group, unfortunately, and the language and culture can be a bias. And recruiting participants was quite challenging, even if it doesn't seem like it was. For the improvements, there are quite a few improvements that we can do with this. The survey design, I think we should focus less on quizzes and more on capturing emotional and behavioral responses. People do not like to be quizzed. People do not like exams. Also, incorporate validated question sets. So there are question sets online that people have been used, and then we could use them to actually compare to literature and see what other people have found with this special question set and use the same ones. Also include a pilot test. I've learned that people do not like my surveys after I've given it to them, and then add post-broadcast surveys that are a bit later. To reach more audience, I would launch an early outreach campaign to reach more people and also introduce a reward program. And for Reefs Go Live, how to improve it, I would really embrace the interactivity because that's what makes it innovative. And also partner with local scientists, educators, bring new people, bring changes to each broadcast. To summarize, we looked at the educational impact between two video formats for children and the general public through pre and post surveys. And we looked at knowledge, emotional engagement, and conservation behavior. And we found that knowledge improved, but I want to say that with the sample size, even if there was a significant difference between the broadcast and the video, with a sample size, it's hard to say that it is the response, the answer, the global trend, that documentaries are better than broadcasts. But also, live broadcasts increase curiosity and engagement, documentary encourage more reflection, and the conservation intention rose after watching these videos. Now I have a question that I think needs to be answered. So how can we inspire 8 billion people to value and care for the ocean, as this will change everything? Thanks to the whole CCMI team, and thank you for listening.
Allez, let's go, Florence. If you're okay, Florence, you can go.
Yeah, of course. Thanks, Luna, for the inspiring presentation. I have a few questions. The first one is if we could go back to page 27 of the slides. You mentioned that there were, is it 360 respondents or answers to the different surveys?
Yes.
Answers. Okay, great. That clarifies my question. Yeah, because I imagine with the fact that sorry.
Here?
Yeah.
Yeah. So I did a total of all the answers that I had for the pre and the post. So yeah, so I counted answers as participants. I expected that people did not answer twice or more.
No, yeah.
Okay.
So it should be participants as well, but it's hard to say since I didn't ID them. I didn't have individuals and knew which one answered what. So it's hard to say. So it would have made more sense for me to say there are answers because some of them filled the pre-survey. Like I said, they were like more than 80% they did. So that means that some of these people are the same people. So that's not 364 participants, but 364 answers.
Okay. And I guess that's counting across the 4 videos as well, where you might have had some repeated participants? Or was it?
Yeah. So that's across all the videos. Yeah.
Okay.
Yes.
Great.
Yes. Sorry.
Thanks for clarifying. Yeah, no worries. And you mentioned that you were, well, actually, no, maybe I'll ask directly why you think the lower sample sizes in the pre-recorded videos occurred, like why there were less people interested in those than in the live broadcasts, or is it the way you communicated on it?
Probably, yeah, because what I did with the participant for the broadcast is CCMI was also a place to host schools. So schools could come directly to the station and they had lessons with the people. There was an educational team and they would bring the kids snorkeling and everything. And so what I would do is I would talk to the teacher and talk about this research, the broadcast, and see if they were willing to participate with me. So I had this opportunity to actually do it with the kids and have, therefore, more people attending the surveys. For the video, I asked a question on the broadcast for them do you want to participate in another experiment that is similar to this, but that involves, as you know, and you can, then they decide to put their email address or not, and then I sent them an email to, so only the people that saw the email and decided to participate were the participants in the video survey.
Would you consider doing anything differently to recruit more, to have more of a equal sample size between the two types of videos?
Yeah, absolutely. So I would do a lot of things differently. But the first one is probably I gave myself a bit too much time before deciding to make the video. And I think if I would have done it earlier, then I would have more chance to use this and organize maybe watch parties on the island, and also use the kids that were coming to the station as well to do it. And then the only problem with it is that there was also some media requirements through CCMI that it was a lot to go through, and because it was a bit late as well, I needed approval, and they didn't want, especially for me, to share the video everywhere. So that was also like, if I would have a bit earlier, then I could have probably had this approval and therefore like go and broadcast it a bit more.
Yeah, fair enough. And then, yeah, maybe just a question on the results. You mentioned that the last video gave different or less positive emotional responses than the other ones. The one on climate change had more concern and worry. Why do you think that is? Is there any difference in the way that you structured the story arc of how you balanced positive and negative problems and solutions?
So I tried to keep it the same sort of format as episode 1, which episode 1 was quite educational. We followed a scientific method of an introduction, methods, results, and then discussion. And literally it was about a research that CCMI had done on seamounts. And then to not have too much difference between them, I tried to keep that sort of structure. It wasn't about a research, especially following that method, but it was with an introduction and really educational without like too much of a storytelling. So I tried to keep that format to avoid that, but still talk about hopeful things and talk about the bad thing, but still talk about the hopeful things. I think what changes is that maybe people are more excited about seeing it live. And that's why they felt like more curious, because they could actually, we had a lot of questions of kids that were asking like, oh, what's that fish? How cold is it underwater? What's the temperature? What's your favorite fish? So I think this ability to ask the question, even though the episode was quite educational, brought that feeling of feeling more curious and engaged and with the broadcast rather than with the video, where you're just passive watching.
Yeah, fair enough. Thanks.
Thanks, Florence.
Yeah.
A couple of questions.
Thank you, Luna, for this nice presentation. So it's a lot of data generalized and collect and so on. So congrats. I have one, I think, big question.
Okay.
So I like the fact that experience physically the ocean environment, what they really get from only video, and what do you think would be next step?
I mean, that's true that, you know, the only way to really learn and to really put value on something is to actually discover it yourself and not watch it as a video. I mean, we still have, with all the video and all the media that we have online, with the social media, we like still end up doing passive watching and not really caring for it. I mean, everybody just scrolls down on their phone, just watching video, oh, that's pretty, and then just keep going. So, I mean, the next step would be really to have them go in the water and make it like, but it's true that it's not possible for everybody, and it's something that would require like a lot of funding. And but really, like, I've seen it with the kids coming at the station, and even there were university groups as well, 20 plus years old, that would come and were landlocked and never been snorkeling before. And I could see that, how it changed in their mind, how they put, like, how they saw it and they found it beautiful and decided to care way more for it and understand. So really, really the way to change that is to make them see it by themselves, but to actually make them do that is very...
Yeah, but then in your video, can you envisage promoting also this specific aspect, in addition to the knowledge itself?
Yeah.
Promoting people to go outside
and do it.
Yeah.
It would
reduce their screen,
yeah,
time, and then go in the water, even if it's river or lake or also marine environment, but at least experience. Yeah, I think because you grab a lot of audience with this podcast and so on, so it's really nice. And I think what more insight regarding your data, it's really to ask this question about all the respondents.
Yeah.
Did you get any physical experience with the ocean at one point?
Yeah.
Because you will get, I think, much more insight for the topic here.
And then maybe like, will you do something about it? Will you plan to try and go and see those? Yes.
But at least if they experience already in the environment.
Yeah. How do they? Yeah. Well, there was
also the answer.
There was a question, was how do you feel connected to the sea? I guess it's quite vague in a way, but they had a multiple choice asking like, I live near the coast, or I enjoy doing these kind of activities.
Nice.
So then I could, something that would look more into it. Okay.
Thank you.
But I think it's a good start, the broadcast, to like bring that interest for those people to actually be like, okay, now I want to go and see. So I guess that's.
Aziz?
Thank you very much for your talk. My concern about this is the David Attenborough stuff. He also says that action is needed now. So not really concerned for it really, but this is obviously for children. How can we kind of generate the same influence on people that are actually making decisions now with this technology?
Yeah.
You can say we can't if you
That would be nice. That would be nice. But what was interesting is that I still had quite a lot of people that were not children that did that watched the broadcast and filled the surveys. And I see it with my parents that obviously supported me. And we watched the ocean documentary from David Attenborough a few days ago, so I could be prepared in case. And they talked about seamounts, and they were so happy about it because they didn't know anything about it. So even if it's aimed at children, people that do not know still seem to learn something from it. And it really made me happy because they were like, yeah, seamounts is this migration step where, you know, species feed and coral bleaching. My friend from the Cayman was telling me how he was telling people on his boat how bleaching works when he didn't know about it. So it still works, but it's true that there's a way that we could make it even more impactful for higher people, higher organization to actually understand it and want to make a change because of it. And I mean, yeah, there are so many ways to do that, I guess, but like I said, in the end, I would bring more educators in it, more people that are experts in it, and really have that exchange and bring more interactivity, less games, but more polls, something more adult than just games.
You mentioned Howell 2011 says, I guess that's for adults, like retention fades after watching a documentary.
Yeah, so that study was on adults that watch documentaries about climates.
So do you have any data on the kids and when they watch a video or this live stream, their retention?
On long term, you mean? So no, so that's one thing that would be good to do with this study, is to actually not look at immediate impact, but actually look at the long term impact of it, and if they actually feel the same and have remembered the same. But again, coming back to my parents, they watched the Seamount episode. I mean, they're not kids, obviously, and knowledge retention seemed to be a little bit better in older people, but they watched the first episode of Seamount in early March, and they were still able to tell me what was, so it kind of worked a little bit. That's what I'm trying to say. But yeah, for kids, it would be really interesting to see the long term, and that is something that I tried to do, but it was really hard to also get the teachers to implement that in their
I just have one final question on a comment you made in your presentation. Traditional knowledge doesn't work anymore. Can you explain?
I mean, traditional. Okay.
Yeah, it's a traditional teaching.
It doesn't work.
She did say it.
That's it. School is over, kids. Let's go.
I really think we have to add more of that.
Can you explain what we do wrong?
That is not what I meant. I mean, I think that teaching should not be about having kids sitting in a class and listening to the teachers talk. That's what I mean. It's the way of teaching that should change, not traditional knowledge. I was probably error on my side, but I mean, MARRES is probably going to be happy that I say that, but the videos, the capsules, although they were quite horrible to watch before, it's a different way of teaching. And I appreciate that about MARRES, to not just be sitting in a class and just listening to someone talk, but that we had the opportunity to learn about the topic beforehand. And I think teaching should be more of a discussion today. It should be more interactive. It should be more of a discussion. I said discussion already. And it should be interactive. And I've seen it with kids when I did some voluntary work a long time ago, my first kind of ecotourism thing. But it was all about, you know, creating those games, and they would learn so much, and they would be so happy about it. And it was little games to understand food webs or things like that. And I think that kind of teaching where it brings innovative ways of learning, especially for kids, is needed because kids do not work like that anymore because of everything that changed. The technology, everything is just changing, and we should also adapt the way we teach. Sorry.
I have a couple of questions for once. Yeah, because it seems you have selected different formats also for the lives.
Yes.
Was it like on purpose, and have you been able to see some differences?
Yeah. So so the main differences were with like the emotions mostly, but apart from that, I didn't see a lot of differences, and it's also because they had different formats, but not as much as I would have liked them to have different formats. But it's true that the first one was way more educational, like I mentioned. The second one was a bit more fun, and it was really light toned. So it was more of a, we were asking kids, like, what superpower do you want to have? If you had a superpower underwater, like, what would you have? But we would compare that to the colors of a scorpion fish, or those kind of things.
I'm sorry, we're smiling because it's not a question we asked to all the future MARRES students in their interview. So.
Oh, what, the superpower?
Yes.
Really?
If you were a fish or.
And I think that it's really important to keep people, not just kids, but their imagination. I think it's really important to keep them creative and keep them.
Exactly. What the question.
Think about these things, and we had much more games in that episode as well, so that's why I was a bit more lighthearted, and people felt way more engaged with that episode. And the third one was on coral restoration. We were showing a lot of videos of coral restoration, how to do coral restoration like during the episode. So that's why I said it was a bit more action driven. And the last one was climate change, and our topside host actually shared his experience of seeing the bleaching event in 2023. He saw the change from really healthy reefs to bleaching to completely dead. And he was saying that story, and he was really sad, actually. But we still brought some, inspiring things, hope after, to just make sure that we're not depressing the people.
Everyone. You mentioned language barrier, and it's not clear to me because I thought it was kids from the US.
Yeah, so kids from the US, but also tried to target a broader audience. And so there were some people from France, and I had some comments. There was a question, what could have been done better? And the equipment, when you're in the middle of the sea trying to find internet, the sound is not that great all the time. And for people that do not speak English or is not their first language, they found it more difficult to understand. So that they couldn't understand everything that was said in the broadcast, and therefore that would be, reducing the.
So you don't have any example of that, a video you want to share?
I mean, it's on YouTube.
It's on YouTube?
Yeah, it's on YouTube.
Because actually it connects to a question from Jacob, which is like, do you think it will be possible to increase reach by streaming on more popular platforms?
I think.
So what's the platform you use, and why is this one? Is it restricted? Do we have to pay?
It was used on the Class C platform. It's a teaching platform where you can create those events. And people registered for it. That's why people have to register, so they can access the event. And
you want them to register so you can target them for the questionnaire.
Yeah, but it's for specifically that website, because for YouTube I couldn't track the number of people registering for the broadcast. But I agree with you, Jacob, that I think it should definitely be broadcasted on platforms that people use,
especially kids.
Especially kids and TikToks, and maybe make not just the episodes, but also maybe smaller episodes in between where we introduce the subject and it's live as well, and just to make some sort of trailer that is live where people can start to ask questions. But yeah, it was also on YouTube. So it was two platforms, YouTube and Class C.
Okay. You have a question? Yes?
I was wondering, like, the kids that went to visit, what is kind of their background? Because I imagine kids coming to the Cayman Islands are not a bit, they probably have some money. Would that affect, you think, the reaction?
So it was really interesting to see all these different type of kids, and also younger adults. So for the younger adults, we had people that came from fashion background, and it was like some sort of course that they could take with a university where they decide to go somewhere, and then they have a field trip to this place. But for the kids, it was a bit of everything. It was, but for the kids coming from the Cayman Islands, I mean, it was a bit of everything. Some were very interested and really wanted to learn more and wanted to get something out of it. And some others were caring a bit less. And you could see that maybe it was because of a different background. Whether values have been taught, I do not know, and I do not know if I should keep talking about that. But it was very different. It was a broad range. But what I saw was a big difference between the kids coming from the Cayman Islands and the people coming from the US, the kids coming from the US. And they seem to, definitely the people from the US seem to enjoy it like a bit more, since kids from the Cayman Islands, they have an idea of what is there, how it works. And even though the island was really different than Grand Cayman,
Talking about kids from the US. Ali?
So your role you were the videographer. Right?
Yeah.
So you were the one that's, when they were streaming the thing, you were the person behind the camera.
So not the one filming the underwater host talking, but I was the one filming images around.
Okay.
And for the last one, I was actually the producer. So I was the one making sure that, what appears on time, change the, the screen. So one is the topsider talking to the topsider. I was also in charge of writing the scripts and making the production plan of what are we filming, when are we filming it, and all of those things.
Yes.
I think all of this is fascinating, and it is really, really important because none of the things that we do would be out there without what you're doing. And I think only taking a photo underwater is sometimes challenging, you know, like the light and all of this. So what you're doing, like not only going from photo to video, but also trying to have an educational purpose, like this is something I personally admire, and I wanted to say that. And that being said, I want to know, I mean, other than recruiting people, you know, for this research, what was the most challenging thing for you in the field? And the internet, of course.
I feel like I had the easiest job in the end, filming, because I didn't have to learn a whole script to talk in front of the camera. But I must say, underwater is not the easiest thing because you have a lot of things happening. You are attached with a cable, you have a full face mask. It's a bit different in a way. You breathe, you're way more buoyant. Sometimes the boat turns around and the cable pulls.
Were you attached to the cable as well?
Yeah, you're attached to the cable. The camera's attached to the cable because everything's connected. And sometimes there was some current, so I was trying to get some nice shots. And also knowing, because it was hard, there was no way for the camera person to communicate with the top side. So to know exactly when we can switch, we were just like hoping that everybody's doing a good job and filming at the right time. And that if actually you cannot film what you're supposed to film, that they can see it and do not project it on the screen, you know, those kind of things. So it was a lot of organization, I think, it was quite, but at the end, it was like, before that, it was quite a lot of stress. It was fun. And then after that, it was a lot of release, which was great. But every time was a good time. It was a good time.
It's amazing. I mean, usually, sorry, if I can ask, sorry, very, very short. From what I've heard, sometimes people take 2 to 3 years only to release a documentary. So for you to be able to do this live, I think it's impressive.
Yeah.
Congrats again, also from some people around. So that was a question. So Nathaniel and Jacob saying thank you for answering the answer. And Andrea, congratulations, Luna. Brings emotion to you. So yeah, so more emotion coming because it's lunchtime, so outside. And so for people online, sorry, the microphone was not working really well. See you tomorrow for Fisheries and Communities. Are you ready, Margot, Katerina, Nelly, Lucia? You ready? Good. Allez, on y va. Thanks. Florence, stay around, please.